The immune system is our most powerful line of defense against antigens. These can be external threats, such as viruses and bacteria, or internal threats, such as cancer. T cells are specific cells in the immune system that help identify and kill antigens in the body. Cancer cells can sometimes evade the immune system, preventing our T cells from recognizing the cancer cells, allowing tumors to grow undetected. Scientists have developed an extraordinary solution that equips a patient's immune system to detect and kill cancer. A receptor called a chimeric antigen receptor, or CAR, is developed in a laboratory and added to the surface of T cells taken from the patient. The newly modified T cells are then returned back to the patient, so these cells can identify and destroy cancer cells. Each therapy is uniquely tailored and modified for every patient. This new technology has led to FDA-approved immunotherapies called CAR T cell therapy to treat certain blood cancers. These new therapies are still in the early stages, but are already redefining precision medicine. Scientists are continuing to conduct extensive new research to fully understand how minor changes in the development of these therapies impact safety and efficacy. Friends of Cancer Research recognizes the challenges in the field and is taking action. With a world-class group of experts, we're working to devise strategies and propose updated regulatory policies that promote scientific advancement. Together, we can advance this groundbreaking therapy to bring better treatments and give hope to patients, families, and caregivers.
Good morning. Thanks everyone for joining us today for what I know will be a forward-leaning set of discussions on the future of cell and gene therapy. A special thank you also to everyone who is joining us virtually, and we also welcome you to follow us on LinkedIn and Twitter. We do have a large online component today, so I want to thank everyone in advance for keeping us on time with our agenda, with your remarks for those of you that are in person in the room with us today. For those of you that are new to Friends, we're an advocacy organization working to accelerate policy change, support groundbreaking science, and deliver new therapies to patients quickly. I hope that will be evident today and throughout all of our policy and research partnerships. Today's meeting is an exciting component of our portfolio of work and exemplary of the technological advancements that are shaping the future of cancer research and care. While we will be discussing in depth many of the exciting scientific opportunities and advancements in the cell and gene therapy space, it's important to recognize that we're in a political environment that at times casts headwinds into the face of science and the progress being made. Juxtapose the volume of mistrust and misinformation about science alongside the groundbreaking progress in research, it can add tension to current policy considerations that have direct implications on the future of biomedical research. For example, despite congressional action last year to strengthen an important program like the FDA's accelerated approval pathway, which is a valued tool for advancing treatments for serious and life-threatening diseases, there is still consternation about its use. If we're not careful, making modifications to address specific situations outside of oncology will have substantial effect on the future of oncology, as opposed to a more innovative approach of leveraging what has been learned through decades of successful use of accelerated approval in cancer to inform how it can be leveraged effectively in other therapeutic areas and across the healthcare system. I want to thank CMS for recently reaffirming their agency's commitment to accelerated approval. These are science-based policies that can make differences in people's lives. I think we'll hear today that the science continues to rapidly evolve, and together we have the opportunity to ensure that future policy keeps pace. Thank you again for joining us. We look forward to a robust set of discussions, and I hope you'll engage with us today and all of the content that you'll hear moving forward. Now, I'd like to turn things over to a major policy driver herself and introduce Friends Chair and Founder, Ellen Siegel.
Thank you, Jeff. You call me a policy driver, it also can make people crazy too. I want to thank the Friends team. You guys are just awesome. The work we do is extraordinary, and it's hard work every day. And I also want to thank all of you who work with us and support us. We cannot do it alone. We do it together, and I'm deeply appreciative. Today, this introduction is very emotional for me because Arie Belldegrun and Steve Rosenberg I have known for, well, Arie 38 years and Steve 39 years. It's pretty remarkable because I was 10 and I really remember it. But they are pioneers in what they are doing. I met Arie when he was working with Steve at the National Cancer Institute. And as you know, Steve Rosenberg is a legend in all of his work he's done in immunology and CAR-T. He's still there and working hard on science with one goal and one goal only, and that's to help patients. So we are incredibly appreciative. Arie went from the National Cancer Institute to UCLA, and he's a serial entrepreneur, a scientist, and has the same goal, making life better for cancer patients. So we are incredibly appreciative. It's very emotional to see these friends who I've known for a long time, way before I was involved in cancer. Actually, it was before my sister died. And Steve was helping me, and Arie did, and so I've known them when I was in real estate, believe it or not. So again, thank you for what you're doing. We look forward to you guys speaking, what you're going to say. Don't be shy. We have 1,000 people on through virtual, and a lot of people here that are interested in what you're saying, and your words have significance, and we're deeply appreciative. Thank you.
Where's Steve? Okay. Thank you. Good morning, everybody. And while Steve is coming in, let me just tell you that no matter how many years passed since your fellowship, your residency, when you sit with a mentor like Steve, you are nervous, okay? It doesn't, you remember the first day that you came in and Steve looked at, and what do you want to do? And well, I was 6 years in residency in surgery. All I knew is to cut, and I felt very good. And while I'm coming to the NCI, I see a group of surgeons like me coming from outstanding institutions, and all of them are infected with a bug called, over 3 decades ago, immunotherapy. All of them are talking about immunotherapy. I said, where is the surgery? When are we? Oh, when we need, we'll cut. But this is about immunotherapy. And what we saw here together, when we say together, that was the environment those days at the NCI. Benches, surgeon next to a PhD studying the in vitro, somebody preclinical, one of them a medical oncologist. We were all in actually one huge room, but everyone interconnecting with the other, learning from each other. So this is the invention of the multidisciplinary programs. It was in that NCI that everybody was sitting there, and at 11 o'clock at night, we all came back to continue our experiments. Those were the days. So when you are looking at the future of together collaboration between academia, regulatory, and clinical, and everyone, that is kind of the most important message I can talk today. And even agencies within the regulatory, everyone needs to talk with somebody else, otherwise things will not move. Well, with that introduction, Steve, thanks for having you here. We started 2 decades later. I came to you, it was 2010, and you said, hey, come to my office, I need to show you 2 patients. Finally, we are treating patients with CAR. I said, wow, let's look at it. And maybe tell us a little bit about the story at that time of, it wasn't so long ago. You remember it very well, those days.
So we began our work using lymphocytes as a drug to treat cancer because of the recognition that the immune system had the extraordinary specificity and sensitivity to distinguish a malignant cell from a normal cell. And so many years earlier, in 1988, we published the first paper showing that, in fact, the transfer of lymphocytes that were sensitized to cancer, in that case melanoma, the transfer into patients with metastatic disease could, in some patients, cause the complete regression of that disease. And those initial observations then led us to try to develop cell therapies over the years. Now, with the advent of CAR T cells, we already had the substructure available for the production of cells under good manufacturing practice regulations to treat patients.
Your mic is cutting out. Yeah, just take this. Take that.
Okay, I guess it was a mic problem. Were people hearing me, or? Well, I'll start very. At any rate, we had developed the substructure to develop cells that we could then administer to patients, their own cells, that could potentially be used to treat them, and in fact showed we could mediate the regression in patients with melanoma, the transfer of their own autologous lymphocytes. Now that almost immediately then led us to develop ways not to take advantage only of the natural cells that existed in the body, but looked at ways to genetically modify them. And in the year 2000, we introduced foreign genes into people using lymphocytes to track these cells through the body, and in 2006 published the first papers showing that in fact one could genetically modify a normal lymphocyte by putting in a conventional T-cell receptor, the kind of receptors that exist on normal lymphocytes, and convert that normal cell into a cell that could then attack the cancer. And when the concept of CAR T cells came along, it was then very naturally for us to begin experiments utilizing those cells to treat patients with lymphomas. And as Arie mentioned, in 2009, we treated our first patient. He had a diffuse lymphoma throughout his chest, his abdomen, lymph nodes throughout his body. He underwent a complete regression, now still ongoing over 12 years later. And in fact, of the first 10 patients that we treated in 2009, 2010, 6 had complete disappearance of their cancer, 5 of which are ongoing to the present day. So it was quite clear that in fact this was going to be a treatment modality of some use. And interestingly, several large corporations, pharmaceutical companies, came to us to see what we were doing, and in fact they would look at the data, and I remember one large company brought in 11 people to go over all the records of all of our patients, all of our procedures, and in the final debriefing at the end of their visit, they said, Steve, if we have this disease, we'll come see you, but we don't see any way to bring this to people at large. And then enter Arie Belldegrun, who had been my friend then for 25 years, since you had been in the lab. And Arie had the vision that, in fact, this could be developed into a treatment that's brought to people around the world. And of course, at the NCI, the National Cancer Institute, what we do is discovery, we do innovation, but we have no ability, of course, to develop and deliver these treatments to large numbers of people. So Arie came in, reviewed our data, and had the vision of creating an organization that could deliver these kinds of findings to the world at large. And he did that incredibly successfully, because we then worked together to refine the treatment, making changes as we reviewed the data on a weekly basis together, until we finally came up with a regimen that was evaluated, and the rest is history. The KITE organization, founded by Arie, then became among the first to deliver this to people, and now is being extraordinarily effective in doing that. So I owe a lot to Arie in terms of having the vision to take cell therapy to people, as opposed to just working on research projects as we did at the NCI.
But what is kind of more impressive or more important for me was to create the cooperative agreement with the NIH, what's called CRADA. And people don't maybe appreciate all of the opportunities that open to us. But the minute we have CRADA, we started working collaboratively to develop CAR-T with no barriers between the NCI and a industry. And I remember our starting. Today, for example, the lymphodepletion, Cy/Flu, it's great. Looks very simple, everybody's using it, the same dose, the same standard, but we had to find out, how do you do that? There are variables. There are variables to the number of cells. Are we giving the high dose cells, 6 million, low dose, intermediate dose? Those, if we would do it purely in a small biotech with very few, with budget constraint, we couldn't have done it. So many variables, but together we said, okay, let's try one variable at a time and do it in few patients, 3 to 5 patients at the NCI, and move on. And we could have moved, from a high dose that was too toxic to a low dose that was not of cells, that was no response, to the mid. So we said, we finally have a dose of the cells. Now we have to find the Cy/Flu. How are we combining the two? This project, which could have taken a year or two alone, happened within 6 months or even less. And then we could start and move to the pivotal clinical trials. So that's kind of an example just on the future. But when we came to the NCI, you were not a great believer at that time in T cells. It was about your lymphokine activated killer cells, which essentially are NK cells stimulated with IL-2. So the first, 12 months of my time, we were working on NK. Okay, now everybody talks about NK, but forgets about how many patients did you treat? 100, 150, as I remember, with NK cells. These are the same NK as from 30 years ago. So what changed, and how come that by the time I left at the end of '88, there's a New England Journal of Medicine about T cells and their role in immunotherapy by our group?
We started out enthusiastic about the use of these lymphokine-activated killers, which are very NK-like cells. We saw responses when administering with IL-2. We did a randomized trial and, in fact, showed that the LAC cells were not contributing. It was mainly the interleukin-2, and, in fact, that was approved by the FDA as the first ever immunotherapy back in we did those studies in 1985, and it was finally approved by the FDA in 1992 for the treatment of patients with renal cell cancer and then melanoma in 1988. But when we did the randomized trial, it turned out the LAC cells were not effective, and I then immediately moved to the use of conventional T cells that have T cell receptors with exquisite specificity for the recognition of their antigens. And it's these T cells that protect us from viruses, from other environmental stimuli. These are the critical cells of the immune system that adapt to change and can develop reactivities that protect us against opportunistic infections, viruses, fungi, and so on. So we had begun studying T cells and their T cell receptors, and those were the cells that then ended up causing regression in patients with melanoma. And when we put those T cell receptors that were unique and naturally occurring and could recognize cancer into a patient's normal cells, those normal cells became anti-cancer warriors. And when we inject them into the body, and this is a very important point because this is a living drug, as opposed to most chemotherapy agents that are excreted within minutes to hours of their administration. When we give these living cells as a drug, they expand 10,000-fold inside the body over the course of the 2 weeks after the administration. And they patrol the body when they see their target that's recognized by the T-cell receptors on those cells and recognize a target on the tumor. They will stop their circulation, they will begin to divide and manifest their effector function of destroying their target. And that's what an immune reaction is. These cells continually patrol the body. Every 14 seconds, they're pumped out of the blood throughout the arteries, come back to the blood, back to the heart, and then circulate around the body again. And so we were taking advantage of natural immune responses with cells that we could identify with anti-cancer activity or cells that we could create in the laboratory by genetic engineering to attack the cancer.
But today, with engineering of NK cells, you can change, enhance, change the structure and the function of NK. Do you think we can bring it to where T cells are? What's your personal view of that?
So the critical observation is taking a T-cell receptor, the kinds of molecules that are on the surface of a normal lymphocyte that, again, recognize foreign antigens, and take those T-cell receptors and move them into a patient's normal cells. So you don't have to do all of the recognition capabilities of identifying those receptors time and again. Now, if you put them into a patient's autologous cells, that is their own cells, then the body doesn't recognize them as foreign and they work. That would be true of NK cells, where you could genetically modify the NK cells as well and probably do the same thing, although we've emphasized the use of a patient's own natural lymphocytes. I don't believe, although there will be controversy about this point, I do not believe there is any credible, reproducible evidence that natural killer cells alone that are not genetically modified with T-cell receptors have anti-cancer activity against established vascularized cancers in the human. I know there's a lot of work going on in that field, but to the present time, natural killer cells are not an effective weapon in this fight.
How about other cells? We have now so many companies in macrophages, iPSC, gamma delta, and so on. I mean, multiple, and each one feels that this is the answer to anti-tumor rejection.
Well, there's no question. There's a need to explore all possibilities based on sound scientific evidence. And so there are gamma delta cells, there are macrophages, there are a variety of other cell types that are certainly being studied, but as of now, I'm not aware of any reproducible data that would lead one to believe that those cells are effective unless they're genetically engineered. And if you're going to genetically engineer a cell, why not just take a patient's own normal lymphocytes and do it? Now, the development of these cells can take a fair amount of time in the laboratory, and I totally applaud the possibilities of taking these allogeneic, somebody else's cells, genetically modify them so you have a drug on the shelf that can be immediately delivered to patients, something that I know Allogene and other companies are working on. And that stands to dramatically simplify and make substantially less expensive the ability to do cell therapy.
Let's move a little bit to the more the technology from the car. Now we have the TCR. You have the TIL that will eventually make it to the clinic. Other modalities that are the neoantigen that we have, that you have put so much effort today. Where are you see the field going as we are moving to our next topic, which will be kind of a solid tumors, how to crack solid tumors? What is the next field that's needed?
So the CAR T cells, just so we all understand the underlying biology, the CAR T cells take a monoclonal antibody and insert it into a lymphocyte and thereby convert the lymphocyte into the reactivity of an antibody rather than the recognition reactivity of the T cell receptor that naturally exists in the lymphocyte. Now, these monoclonal antibodies were first described by Kohler and Milstein in 1985. So we're 48 years into studying monoclonal antibodies, and there's yet to be a single monoclonal antibody described that can uniquely recognize a cancer without also recognizing normal cell components. And there's a lot of confusion, therefore, that surrounds this. You can only develop a CAR T cell if you have a monoclonal antibody that's specific for the cancer. And they largely do not exist. There are a few very rare examples, but they're very poorly expressed. And I do not today see the application of CAR T cells to the solid cancers without some new innovation that today we're not aware of. We need to realize that it's just as easy to kill a normal cell as a cancer cell. And if you have a receptor or a CAR T cell that can recognize something on a normal cell, that normal cell can be destroyed. And we and others who have worked in this have resulted in the deaths of patients by giving reagents that could recognize normal cells as well as tumors. And so in the absence of a good monoclonal antibody right now, I don't see how CAR T cells are going to be effective. The problem is the targets. Now, conventional T cell receptors can recognize 5 or fewer molecules on the surface of a cell. And it's these T cell receptors that I think hold the hope for the ability to treat the solid epithelial cancers. We need to realize that over 600,000 Americans die of cancer each year. 90% of them die of the solid epithelial cancers, all the way from the rectum up through the stomach, the esophagus, the ovary, the prostate. Those are the killers of 90% of all people who die of cancer around the world. And I think it's using these T cell receptors that are exquisitely sensitive, can recognize a single amino acid change in a molecule and attack it. Now, CAR T cells have shown effectiveness in the hematologic malignancies because the normal precursors, the B cells, get wiped out, but you can live without B cells for an indeterminate period of time by having gamma globulin infusions. But the precursors of these B cells are not expressing CD19, and they subsequently recover over 6 to 8 months. The solid, and so you can use CAR T cells, even though you wipe out the normal cells, you can live without them. If you wipe out the normal cells in the liver, in the lung, in the pancreas, people cannot survive. And so there's been substantial progress made now, not in using so much CAR T cells for the solid tumors as T cell receptor transfers, either naturally occurring cells or cells into which you can put T cell receptors that recognize the solid tumors. And we've seen regressions in patients with colon cancer, with pancreatic cancer, cervical cancer, bile duct cancer. The examples exist. It's not, does it work? It works. The problem is that it doesn't work well enough, but there's enormous effort ongoing now around the world to try to improve upon the use of conventional T cells transferred into normal cells that can then become effector cells to treat cancer.
Last question, I'm just looking at the watch. Accessibility. Today, 1 in 10 patients are getting therapy in CAR T. We never thought that the CAR T would be so effective when we developed. That's kind of the facts. But they have been, and they're really working very well. What's next? How do you make it available to the masses? What's the accessibility? Because I know we'll talk about it the next session. But in your mind, how do we make it available to everybody else?
The problem is the personal nature of the recognition of the cancer. We studied 205 consecutive cancer patients with whole genomic sequencing to try to identify immune cells, T cells that could recognize those cancers. And 75% of the time, regardless of the histology, we could find them. But the T cell recognition was almost always unique to that patient. And so in those 205 patients, we identified 365 different antigens. But 364 of them were unique to the individual patient in which they were found. Only 2 were shared among patients, and that turned out to be KRAS, a very common oncogene. And so the problem is the personal nature of the recognition that's needed. Now, we know that by identifying an individual's own T-cell receptors that can recognize the cancer, we can develop an effective therapeutic. But it's that highly personalized nature that makes it so complicated in terms of widespread application. So there are two problems. One is that technology has to be developed in a way that it can be done quickly. And now we can sequence the whole human genome in 2 weeks, including all the bioinformatics, and do that for minimal cost, several $1,000s. So the technology exists, but it does take time to develop. It takes expertise in the laboratory, and it's going to take the kind of vision that you had for CAR T cells to take this kind of technology to people at large. Now, one of the problems, unfortunately, in modern medicine is that the convenience of administration of a treatment often supersedes the best of clinical judgment. And when we first developed interleukin-2, which could cure patients, about 10% of patients with renal cell cancer and melanoma, because it was difficult to administer, it never achieved the kind of distribution that it needed. And I fear that that's going to happen with T-cell receptor T cells unless we continue to innovate, develop the technology, and convince physicians, oncologists, that it's more important to find something that will cure the patient than will be simply easy to administer.
But while we develop all of that, essentially we need off-the-shelf therapy. That's kind of the idea. Otherwise, it would be complicated.
Well, off-the-shelf therapy, when you have a common target, is quite straightforward. And so for CD19 and B-cell malignancies, that target is shared, and especially if one could use allogeneic cells that were genetically engineered, it could be an off-the-shelf reagent. It could be delivered by a pharmacy very quickly. But when each antigen is unique, it makes it more difficult. Now, there are, fortunately, a couple of mutations that can be targeted that are common. KRAS in about 30% of patients with pancreatic cancer, P53 in half of all cancers. And so we've actually developed a library of those T-cell receptors that can be genetically introduced into a patient's own cells in much the same way that CAR T cells can be introduced. And we just published the first paper showing that we could actually do that in patients with the solid cancers. So this is a field that's very much at the forefront, in my view, of what we need to be emphasizing in the development of these new cell therapies. That is the personalized cell therapy that is going to require T-cell receptors rather than CAR T cells, at least given our current state of knowledge.
Well, thank you so much. We could have continued to discuss and have fun, but the red light is blinking. Thank you all for listening. Thank you.
Thank you very much, Arie and Steve, for laying out the promising future and the challenges that lay ahead as well. And we're really grateful for you joining us today and for everything that you both continue to do and lead. Next up, we have an exciting session entitled Designing a Risk-Based Approach to Support Data Extrapolation Across Product Versions. And I'll invite the first panelists to start making their way up to the stage. The next two sessions that we have scheduled will largely focus on concepts and proposals that were described in the white paper that you received in advance of today's meeting. That paper was constructed through the work of a multi-stakeholder group of experts, and we very much thank them for their time and expertise that they've shared with us. Throughout today's discussion, there will be an opportunity for questions and answers with the panelists. I would invite those of you in the room to use either microphone in the aisles at the back of the room, and for those of you that are watching virtually, to utilize the Q&A function at the bottom of your screen. I'd like to turn it over to our moderator for this next session, Patrick Hanley, Chief and Director of the Cellular Therapy Program at Children's National Hospital. Thank you, Patrick, for joining us today, and welcome to the rest of the panelists as well.
Wait for one more chair. Thank you very much. All right, as we're filing in here, it's great to follow such pioneers as Dr. Belldegrun and Dr. Rosenberg. And, it's no small task. Pleasure to introduce our panel here today. So first, we have Dr. Mark Better, who is a CMC consultant. We have Dr. Christine Brown, who's Deputy Director of the T-Cell Therapeutic Program at City of Hope. We have Dr. Vicky Coutinho, who's an independent advisor at the moment. We have Dr. Michael Kalos, who's the Managing Director of Next Pillar Consulting. And we have Dr. Ingrid Markovich, who's the CMC Policy Lead at the US FDA. So welcome everybody and thanks for participating in this panel. I'm going to kick off this panel with the first question, which is what types of modifications and iterative improvements are we seeing in the next generation of cell therapies as we learn from current experiences and try to bring these therapies to additional patient populations? Dr. Brown, I'm going to start it off with you.
Thanks, Patrick. And I think this is a great follow-up discussion from Dr. Belldegrun and Dr. Rosen's introduction and history, which was just so inspiring. You know, it's an incredibly exciting time for cell-based therapies. And I think this really builds on all the technological advancements that are ongoing in the field and that we see, and it really inspiring sort of intense complexity and novelty that we really want to bring to the patients to really see and really achieve these therapeutic outcomes, particularly in solid tumors. So when thinking of these advancements, I think we'll talk today as a panel about the gene engineering advancements, the ability to engineer these cells to express multiple gene targets under different regulatory elements, and also multiple gene editing technologies for gene knockout, gene knock-ins. So what's going to, what we're seeing are these cell products that not only can target a tumor, but can potentially modify the tumor microenvironment, as well as, provide molecules that improve persistence, fitness to these cell products. And so as we work to iterate these cell products and build in more complex networks for both control, potency, it's going to be really important to build on prior clinical data, but also figure out how we can bring these to patients in a more rapid manner and really be at doses and trial designs that we can really start or accelerate effective cell doses for these patients.
Thank you. Dr. Kalos, do you have anything to add? So it turns out that it's my microphone that's the issue. Just me.
You want me to speak?
Yep.
All right, great. Thank you to the Friends of Cancer Research for hosting this and organizing it. It makes a difference in how we're moving forward our cell therapies. So I believe we're at the beginning of the golden age for cell therapy. And that's a big thing to say, but I really believe it. We've had unprecedented success in the clinical space in liquid tumors with curative responses. We've developed technologies that allow us to knock genes in, knock genes out, knock genes down, deliver genes into immune cells virally, non-virally. It delivers cells deliver and engineer cells beyond T cells, NK cells, gamma delta cells, macrophage, you name it. And do the engineering in a stable or transient way through, for example, RNA. So a lot of pieces of the puzzle are there for us to act on the early successes. We understand through the current successes and failures fundamentally at least some of what matters for cells, the ability to expand, to proliferate, to persist, to develop the area under the curve, to deal with the suppressive microenvironment. We have insights about all of that, cytokines that we didn't have in the past. And we have clinical data that actually informs us about what really matters, right? So this is empiric data. Learnings from the early clinical trials allow us to say, well, here's an attribute of cell that persists that seems to really matter, right? an integration, a knockout of gene TET2 is the prototype example. And we also have empiric data generated preclinically from the high throughput screening approaches that people are engaged in. So we're no longer just testing our favorite hypothesis. We're learning from the clinic and doing the scientific method in toto, right? Look at, identify, and then test the hypothesis. So collectively, we're at this unprecedented inflection point that will allow us to, we have everything at hand to develop the next generations of successful cell therapies. How to act on that in the most effective way is what we're talking about today. So I'm excited to be here.
Thank you. Dr. Better, do you have anything to add?
Yes, thank you. I think Mike and Christine did a good job of explaining where we are, and there's not that much more to add, but from my perspective, we've been working on these engineered cell-based therapies now for quite some time, certainly more than 10 years in a number of different contexts. And the initial premise that you could take a T cell, for example, introduce a new protein into that T cell and that would have a biological effect, it would kill a target cell, has proven to be effective, but there are still many challenges. And so investigators and sponsors these days are looking for more sophisticated solutions to approach the challenge of treating cancer patients. And that may include things like introducing another protein into the target T cell so that it can, say, knock down expression of an endogenous protein in the cell with shRNA, or co-expression of another molecule like a cytokine or another cell activation protein, which isn't directly linked to the killing effect of the CAR, for example, or the TCR, but might enhance the activity of that T cell in an individual. We're also starting to think about how you put switches into cells so that you can only target T cells or target cells that express more than one antigen at a time, so that you can perhaps not be so directed towards non-tumor cells, or you might put a switch into a cell that says, I want it to express this, but not that. So as we evolve through iterative learning about what's effective in the clinic and what might be a good opportunity for the next generation product, we're continuing to get more sophisticated. Many of the engineered cells now that sponsors are working with contain multiple transgenes, 3, 4, 5, perhaps more in the future. And that really gets to the interesting point from the perspective of we're trying to evolve the medicines so they're more sophisticated, but at the same time we need to be thinking about the analytical techniques that will really inform us about what the biological activity of all those molecules are in the target cells. And we're going to need more sophisticated tools and techniques to really understand what our molecules are in the future. So with the scientific discovery to make better molecules, we also have to invest as a community into better tools to really understand what these molecules are and how they're going to work to both improve the outcome in patients and also ensure that the products are safe.
Thanks, Mark. I completely agree about the analytics. I think it's a great point. Dr. Coutinho, do you have anything further to add?
I agree with all the panelists and what they've said today. And what I think also would be very exciting for the field, I know several developers are working on trying to redesign CAR T cells or other cellular therapies so that we don't need to use the chemotherapy preconditioning regime. So that would allow for broader access. It's a very complicated situation to address, but I hope that we can actually emerge through the field with this evolution. And just having worked on both autologous cells and allogeneic cells, maybe what has worked in terms of the first, second, third generation co-stimulatory domains for autologous cells is not necessarily the best place to then overlay for allogeneic cell therapies. So this field also needs to evolve to use better ITAMs or co-stimulatory domains or armoring strategies that would benefit then the innate cell therapies to deliver that efficacy, hopefully.
And so to kind of build on this conversation a little bit, I'm going to take the contrarian view and say, is there a need to kind of keep things simple and elegant? You know, we do have an immune system that works the majority of the time. Are we trying to do too much by supercharging, creating sinks that are, either sinks or turn in a negative signal into an activating signal. Is there something to be said for that, or do we need all these different genetic engineering modalities?
So maybe I'll speak to that a bit. Coming from the academic setting, and most of my work has been in primary brain tumors, glioblastoma, where therapies are desperately needed for this patient population. I think the question is, how complex should we engineer these therapies still needs to be addressed. And we don't know sort of that minimum product that's going to be effective for solid tumors. And I think it's, really the responsibility of all of us to figure that out, right? In really innovative and trials, and really looking at the biology, understanding the cell products, and particularly understanding when you deliver those cell products, what cells are mediating outcomes in patients that show positive response. And so in understanding that, I think we'll get to your question, or the question of the field is, how complex do these products need to be? I think there's a real potential to engineer these products to engage host immunity. So we do want to engage that neoantigen response and bring in other arms of the immune system. And can cellular therapy in solid tumors do that? And that potentially could be an area.
Just to add a couple of comments, right? I mean, I think in the context of an established malignancy, that equilibrium has been set up against the immune system, right? And there's multiple mechanisms through that have evolved to that. So I think the upper limit for success there may be something along what we're seeing for TIL today, right? 30%, give or take. That's 70% of the patients, and more if you count the curative potential that aren't being treated. So I think the future is synthetic biology and engineering of cells. How much is required, we don't know. It might just be a single locus, right? And the prototype for that might be TET2, right? Disruption of TET2, an amazing clinical response in a leukemia, right? We'll get there, but I think engineering has to be part of the solution.
Yeah, and maybe the innovation is how, or the simplicity is how that is done in a more innovative way. Maybe that's the elegance of it. Okay, moving on to the next question. This one, Dr. Markovich, is aimed at you. As we continue to see advancements in regulatory policy, particularly around building upon prior platform knowledge, what opportunities does FDA see for leveraging data and evidence from other product versions to help expedite development and review?
Well, thank you, Patrick, for that question. Can you hear me? Yep. Okay. Well, first, I just want to acknowledge, thank you so much for this wonderful invitation, and many thanks to Friends of Cancer Research for organizing this opportunity to bring all the stakeholders together. This is indeed a very exciting time, and I think as well for folks at Center for Biologics. And I would like to acknowledge the outstanding leadership by our Center Director, Dr. Peter Marks, who's going to be here in third session. So I do hope everybody sticks around to hear some of the thoughts he has to share. So it may help to start by saying there is really no one size fits all when you kind of are crafting policy, as the amount of prior knowledge could really differ from product to product, the amount and the nature. And so for that reason, it would really be very helpful to have an approach when each product is assessed on its own. And a good starting point could be sort of taking a step back and looking at what are the commonalities, what are sort of the common data that have been collected, as well as what are the differences, and what is, to the extent possible, what is the impact of those differences that we observe. Some of the sort of really quick examples where one could look for opportunities to leverage could be, looking at the prior data and using that to inform yourselves to set preliminary acceptance criteria, for instance, for the next generation product. And since part of your question was really specific about the prior platform knowledge, FDA has actually working very hard at implementing the platform technology provision, which most of you is probably familiar. And I actually have a language, so if you would bear with me, I would be happy to read that, because it's vetted through many levels of the FDA. I just want to make sure I get this right. So the platform technology provision is part of the Consolidated Appropriations Act of 2023, and we believe platform technologies can play an important role in allowing developers to leverage information to support multiple product applications. A successful platform could accelerate product development, bring manufacturing efficiencies, and also streamline FDA's review. And as part of implementing this provision, we're aiming to publish a draft guidance by the end of this year. However, we cannot talk about the specifics of this guidance until it's finalized. And with this in mind, we encourage sponsors who are seeking to leverage data outside of the platform technology provision to engage in discussion with the FDA to discuss their specific product.
That's exciting. I look forward to seeing that at the end of the year, hopefully. Okay, and I also want to point out, Ingrid, that I completely agree with you that there's no sort of cookie cutter solution. I do want to point out that the white paper that Friends of Cancer Research has put out, and congratulations to Mark and everyone else who's been involved, they do give very concrete examples of the things that we're talking about here in that white paper. And so while there's differences between the different platforms and products, there's things to be learned from those examples. So well done with that white paper. Okay, so the next question we have for the panel is, when leveraging data from other product versions, it's important to characterize the quality of the existing data. For instance, there could be variability between product batches, especially early in development where release criteria are wider, but tighten as development progresses and clinical data mature. So the question is, what are some best practices for generating high quality and useful data that provide sufficient product characterization and can support data extrapolation for future product versions? Are there commonly encountered challenges that developers face, and how can these be avoided? Mark, I'm going to start with you.
Yeah, thank you. This is a very interesting question for me. So early in clinical development, the time when sponsor will file an IND, it's be very common that there's only limited data available about the product characteristics, clinical manufacturing history, et cetera. But once sponsors get into the clinic with their product, it's incumbent upon them really then to start filling in the details about process robustness for manufacturing, analytical characterization of the product, and all that goes hand in hand with the clinical development of the product. And in cell therapy, it's really unique, I think, in the sense that the CMC side, the manufacturing piece, the envelope around that, is so intimately tied to the clinical application of these products. And the physicians and the people in the development groups and manufacturing and release teams have to work very, very closely together. So all that information grows, and you start learning a lot more about your product as you get going. And then that information that's available becomes the basis upon which you could consider extrapolating data as products evolve. Sort of as an aside, but I'll talk about this because I think it's illustrative. I'm a CNC consultant, and one of the most common questions that I get asked by sponsors, relatively inexperienced sponsors that may not have a lot of experience with their products yet, is, I want to introduce this change. What will be required for me to get regulatory agreement with my change? And that change could be, a process improvement, analytical method change or improvement. It could be a raw material issue. We want to go to a new vendor. We want to modify a viral vector. We want to, you name it, a gene editing tool that we want to change for introduction into our manufacturing process. Or it could be even a new product variant. We think our product will be more effective if we include another transgene, for example. And I always kind of look at that question and turn it back around to the sponsor and say, well, think of it another way. I think the question you should be asking is, what data package would it take for you to satisfy yourself that you understand your product well enough so that you can actually introduce that change and convince yourselves and others that you're not adversely impacting the product quality in any way, and that you're not increasing the risk to patients in any way. And I think to me, that's really the question we should be asking when we think about introducing any kind of a modification to our process or our product, or thinking about a second generation product. It really then brings the question of, what tools do we have to analyze the product carefully so that we understand each and every aspect of it. And as the products that we work on become more sophisticated by introducing more than one transgene, for example, using gene editing tools rather than viral vectors, the number of questions change that we need to answer to really satisfy ourselves that a particular process change or any kind of change, or even evolution to the next generation of product, is really appropriate. And there are a lot of analytical methods that are fairly standard for some of our cell therapy products. We all need to demonstrate that our products don't contain mycoplasma, that they're sterile, that we understand the copy number of the genes that we've introduced into the target cell, VCN, et cetera. But the certain tests, like the identity tests to make sure that all the cells are the same and what we expect them to be, or more specifically the potency tests that we implement on our products, these are more product specific and unique. And as the level of the products become more sophisticated, we may need multiple tools to really analyze carefully the potency of our products and understand how the introduced genes interact with each other. And so at the end of the day, I think the critical aspect is to really understand your product, have well-defined in-process controls during manufacturing, have really meaningful specifications that reflect the true biological property of your product, and ensure that you have a consistent product from lot to lot so that you can ensure patient safety, because at least for me, that's the one thing that's most important that sponsors really focus on.
Yeah, I think analytics comes up again, right, with the product characterization. Michael, do you have anything to add?
Yeah, happy to speak to that. And my experience here is informed by the two academic labs that I established at Penn and prior to that at City of Hope. And in both of those labs, I focused on two concepts: opening the aperture for what we're measuring wide, because what we think is happening is most of the time not what's happening. So understanding what's happening and to our cells in our patients is fundamental. And establishing really elements of objective quality, right? And those types of, that paradigm has informed those labs. It ended up developing concepts like TOSI. We didn't imagine that IL-6 was up in our first patient. We observed it by having a wide aperture and saying what's happening to cytokines, right? And subsequent observations from the clinical space and trials of Penn. So wide aperture, establish assays that help you understand what's happening in the patient in response to therapy. We're now not talking about CMC, we're talking about analytics on the patient, but you can also apply those preclinically and understand your drug. And build out objective quality. And that means that you have to be able to, the data that comes out of your analyses have to be quantitatively sound and understood, right? If you do that, then you understand what your drug is doing. We knew that we had 10^5 copies per microgram expanded CAR T cells in the patients because, by gum, that was the number. It wasn't off by a log or two on the flow, right? And it allowed us to understand how much persistence you needed in patients to have a complete response. So it has to be quantifiable. It has to be referenced. So in the context of what we're talking about here today, iterative trials and how do you leverage information you knew to develop the next, you have to have a common reference. What you're saying is 10^5 has to be what somebody else says is 10^5 too. Otherwise, it really falls apart, right? So how do we build these internal references so that we understand our drug in the same way that the successive product will be understood? And then the third component, and this can be built in academic labs as well as in startups, as well as in biopharma, if you have the right mindset, especially up front, right? Document. How do you establish a documentation process so that all of this information that you're capturing can be captured, transferred, vetted, and believed in as we're moving forward? So in my view, those are the two components, aperture and quality, that allow us to move forward.
Yep, and I want to add, I think it's important that we, to the extent we can, put this data out in the public domain, what are these product attributes that are important? What are we measuring that we find makes a difference? And let's get it out there. And again, the white paper, I think, is a good example of that, and let's continue to do that.
And Patrick, maybe just to add on quickly to this, knowing that these are living drugs, they're complex materials, and they're heterogeneous by nature, CD4 versus CD8 component when you're dealing with T-cell therapy. And we're learning by understanding these trials. So I think Michael brought up the TET2 example where lentiviral integration taught us about epigenetic regulators and how maybe they can improve the therapy. The NY-ESO example of a TCR engineered T cell that was also knocked out, had a knockout of the PD-1 gene, and not seeing an enrichment of engraftment of those cells teaches us something. More recently, the publications of CAR Tregs for patients that might not be responding as well in lymphoma. And so, we want to characterize these products, we want to know what we're delivering, but at the same time, we need to learn by advanced immunomonitoring what are those, maybe aspects of the product that we didn't quite appreciate, and bringing that all together. And I agree, maybe standardizing some of this advanced immunomonitoring and providing that at large to the field.
Yeah, thanks, Christine. Ingrid, anything to add?
Yeah, so this is a really excellent point, and perhaps sort of along the same lines is really looking sort of how do we build more consistency, how do we bring more consistency into our products, and to that end is really understanding the sources of our variability. Understanding is variability coming, for example, from a starting material, from a raw material, or perhaps from the process itself. It could really help with designing the appropriate manufacturing strategy and sort of maybe even swapping the highly variable reagent or building some boundaries around those variable attributes. And additionally, sort of looking at, how do we build that robust product knowledge, that could really serve us very well in when we try to leverage information. Looking at additional characterization studies, which can really go a long way when they're added and complemented with lot release testing, as well as looking at orthogonal test methods that are looking at different attributes and kind of all go towards that idea to build more robust product knowledge. When it comes to asset qualification, since that kind of comes up a lot as a question, generally, we've learned it's better to qualify those assays early in the process, especially if they're being used for additional studies or for comparability studies, because that usually comes up as changes are necessary and required usually to optimize the process. Comparability kind of comes a question. And along the same line, one could also consider looking at the range of attributes that have clinical benefits, and as well as generating more than one product lot, even if single lot can be sufficient to treat a study population, and with the idea that that could really give you a good perspective, how much product variability can your system tolerate while you're still getting the desired clinical outcome.
All right, thanks, Ingrid. Let's move on to the next question. This one, I'll start with you, Vicki. What are the potential risks and uncertainties associated with data extrapolation and how they should be acknowledged, controlled for, particularly if it will be used to justify trial designs and data collection?
Thanks, Patrick, for that wonderful question. So I think in this field of living therapies, as we're talking about today, the non-clinical studies that we put forward are still not really mature to inform or predict what will happen in clinical settings in humans, unfortunately. And so whilst we can generate that data, it can't provide that guarantee around the safety profile of these therapies in humans. So when we're looking at data extrapolation, I think we need to look at the stage of the development of your assets. So in the paper we've talked about maybe your primary or your reference or your first product. And if that's fairly early on in clinical development, then that risk-based extrapolation approach maybe becomes more arduous. you don't have much data around what Mark was talking about, around your specifications, the robustness, the fitness of the cells themselves. So you can't really inform that so well. So you may be incumbent on defending your justifications to the FDA with limited literature, different class products, and also other CAR T therapies as well to date. And so in terms of controlling that information, I think you still have to be cautious with your clinical development and designs, and you can't break the rules per se from those first-in-human strategies, because we don't understand the dose as well, especially if you're taking something into the clinic that's particularly novel. And dose-limiting toxicity periods would have to be observed to preserve patient safety. Intra-dosing intervals as well, we'd probably have to include as well. So there's maybe not much leveraging that can be extended to the clinical setting. However, if we're looking at more mature assets that has generated clinical pivotal data or post-marketing data, then you have more robust information around the critical quality attributes, even the information that you can take, as Ingrid was saying, from extended characterization assays to then make a more informed decision about the risks you could take to maybe then abbreviate those clinical trials for your next iteration advances. Maybe you don't need to start off with the ultimate safe dose that's log loads level lower, and you could justify going in with a more efficacious dose based on the primary or the reference product that you've used. And I'd like to caveat everything I just said, because you could have a CD19 CAR T therapy product made by one developer that behaves very differently clinically to another developer. So you really have to pull together the multidisciplinary approach to bring those stakeholders together to then inform the risk-based approach that we've outlined in the white paper.
I want to point out something you said, Michael, as well, in that we don't necessarily have standardized and hopefully we have qualified assays, but between labs they may be different. And so that in itself is a risk in terms of analyzing and extrapolating from that data, because it will be different from the different groups. Maybe we need to, I think we've talked about this, right? Like one major consortium where we try and standardize that or look at the comparability between them, similar to what CAP does, I think, for flow and things like that. I don't know if you want to comment, but.
That was tried a few years ago as part of the Cancer Immunotherapy Consortium, and it was illuminating in many respects. Labs, variability across labs was remarkable, including labs that were CLIA certified, including labs that were the top in the field. It's a tough nut to crack, especially early on where everybody wants to do their own thing.
Yep.
But we have to find a way to crack it in order to be appropriately informed from others' data, right? And I mean, part of that is build in quality, establish a reference that others can reference too. I think that certainly helps.
And if I can just chime in there, I think, standardization is certainly something we think we could really help facilitate and advance this field. And, obviously this is not something that one person or one company can do. This is really a collective effort. And to that end, actually, CBER does have a standards recognition program for voluntary recognition of consensus standards. And so I'm hoping that you guys are familiar with that. There was a guidance that was published in the summer of last year. And for those who commented, we are very, very grateful. We always appreciate the feedback. But the idea is to really bring the stakeholders together to develop those consensus standards. And this is perfectly, completely voluntary. And we recognize there's a number of benefits for the developer, but also it could really help the regulators bring greater regulatory predictability and efficiency as well.
Great, I'm glad we can give that some visibility. All right, the next question, Christine, I'm going to look at you. What criteria or factors could be used to assess the risk, including what would be examples of a high versus low risk modification? Do you see scenarios where data extrapolation would likely be infeasible?
Right, so really assessing, low risk versus high risk, and then how do you act on that clinically from a regulatory strategy, from a clinical protocol design. And it's really critical, right? Because you want to build and propose a strategy, knowing your product, knowing those critical quality attributes, knowing what you've seen in the clinic. And obviously, we've talked about this on this panel, there's no one size fits all, and you really have to look at the totality. But I can give you some examples from our studies in glioblastoma where we're regionally delivering these cells into the CSF or the tumor resection cavity. And I will say the first time we went to clinic to test a second generation CAR delivered locally into the CNS, that was high risk, right? Very high risk. So we started with a mouse dose. And we used opportunities for intra-patient dose escalation and really strict stopping rules so that we would not be dosing on a toxicity. And we were, the FDA was a partner in this because there were discussions back and forth whether the first patient should only receive a single dose. But given that there's no effective therapies in the recurrent setting for GBM, and we didn't think a single mouse dose would be enough to learn from these patients of how to move forward, the FDA partnered with us and let us move forward with a multi-dose strategy of delivering these cells weekly. And then fast forward, then, I think maybe 8 years later, where we're evaluating a very novel CAR that hadn't been tested. We could build off that data, even though it's a different CAR, different target. We could build off that data, providing that to the FDA, and again, high risk. In between those times, we made manufacturing changes that were critical to improving product quality, product fitness, and working with the FDA, we could repeat doses that we thought were effective, so not having to dose escalate. So these are all examples. So low risk to me would be having that clinical data where you can really do detailed comparative studies that are meaningful so that you know how to build that protocol. High risk would be these novel changes we're talking about where you're using new CARs, new gene modifications, and you're inferring, but you can still use clinical data to help inform and build that best package. So you're going to the FDA with saying, I believe in this and I believe in the dose that we're putting forward.
Yeah, great points. Vicky, do you have anything to add?
Yeah, I think we've talked a little about this. So specifications are useful, but early on, they're probably not so informative. But I think we're talking about, Michael mentioned, opening the lens a bit wider. So there's nothing to restrict us from collecting more information at the specifications stage for information only, to use that to inform for more robustness in later life cycle development, and the critical quality attributes as well can be used. However, they're just informing a good product quality. We don't know if that translates into clinical safety and efficacy necessarily. So this becomes more challenging for us to use the CMC criteria. We've also laid out some examples in table 3 of the white paper that would fulfill CMC data extrapolation strategies that may be useful for developers as well. And then clinically, we can use the expansion kinetics of CAR T cell therapies or other innate cell therapies, biomarker strategies to really inform then how you can extrapolate that data to be more meaningful than when you're going to the FDA or generating your next generation therapies as well. In terms of low risk versus high risk, I think you could take as an example the CD19 autologous CAR T therapies and then adding a CD20 CAR that has been already used would be low risk for the patient. You could justify that with the regulators as long as you're preserving the same manufacturing strategy, the same viral vector. So there's a lot of accommodation for extrapolating the data from also a non-clinical and CMC aspect. However, you will need to repeat a lot of the non-clinical data because you're adding in a totally new CAR as well. So you can't completely get away from doing the non-clinical work. In terms of high risk, fully agree. Then when you're starting to take, let's take the CD19 CAR example, and then maybe you're putting that on an allogeneic therapy, or then limiting through knockouts to then manipulate the autologous cells to become an allogeneic therapy, there comes risk to the patient. If that is not fully guaranteed, then you're going to evoke graft versus host issues, and so that would be more high risk to the patient if you can't prove that success and translate that into a meaningful safety context. And then we're seeing the explosion of mRNA in vivo editing CARs, and that also would be a high-risk manipulation where the preclinical package and even through CMC accommodations wouldn't exactly enable a strong, robust data extrapolation approach.
Mark, did you have something to add?
I just wanted to add one very brief point here. You know, the premise of the white paper that we've drafted is that you can accelerate drug development if you're able to extrapolate data from one product to another, and that we recommend that that be done through a risk-based analysis, as has been articulated here quite nicely. But you know, from my perspective, this whole envelope of information, this collective product knowledge, may allow us to reduce the risk of a process change or a molecule change through much more sophisticated understanding of the molecule itself. And that using risk-based tools is really a good way of assimilating the total information and then determining whether you have the information you need to go to a second generation product, because in some cases you may not.
Thank you. Ingrid, next question is for you. Even in the context of data extrapolation, what additional data is likely always going to be needed for regulatory approval or clinical decision-making when developing a next-generation product?
So this is certainly an excellent question, and I would just start off by saying stay tuned for the platform technologies guidance. We really hope that would bring more clarity to this field. I mean, the usual statutory regulatory requirements still apply, namely meeting the standards for product quality, safety, and efficacy is still pertinent. And as we mentioned before, and I think a number of panelists also echoed this, it would be very difficult to apply one-size-fits-all approach, you know, because the level and nature of prior knowledge could be very different. So if you think about it, the regulatory framework would really kind of have to accommodate these different scenarios while still ensuring that the statutory regulatory requirements are met. And in some ways, you can also really give a lot of credit to people who designed the regs. I mean, they were extremely forward-looking, which is very impressive, because there is still opportunity for innovation while ensuring that all the regs are being met.
Thanks, Ingrid. I just want to remind everyone listening virtually, we are accepting questions. This particular panel is about CMC, and so if you submit other questions, we may defer it to later panels. So thank you. Next question. Are there any barriers, gaps, that might hinder the use of data extrapolation? What technological advances might help to overcome these barriers, and how should we be thinking about incorporating them moving forward? And I know that we touched on a number of these already. I don't know. Michael, if you want to start.
What are the barriers to data extrapolation and what technological advances might we consider? I didn't think about that. Was that one of mine? I didn't really think about it. Apologies. You know, the technical challenges, we talked about it, right? The lack of the ability to directly compare from trial to trial, from product to product in a way that's meaningful. I think if I think about technological advances, we now have platform capabilities that can generate a lot of data, and I think leveraging those appropriately would be part of the solution. I think AI is is everywhere, and AI is, of course, here also, right? So how can we use AI to help us understand and directionally give us a path forward? And then how do we use the AI across products to be able to inform us about? That's the best I can go with.
Well, you know you're good when I put you on the spot and you have a great answer, because yeah, you were not on here. Mark, did you want to answer?
You know, I think we're in an incredible time. There's literally hundreds of products being developed through clinical investigation in cancer patients, autoimmune immune disease patients, et cetera. And as these products become more sophisticated, we've talked about this a little, the ability to really understand through potency assessment, identity assessment, functional activity, these molecules, this becomes very difficult. With a standard CAR T cell, maybe you could look at gamma interferon expression when the T cells see their target antigen on the surface of a tumor cell. Maybe you could look at cell killing, but as these products become more sophisticated, I mean, you can't use necessarily those kind of potency methods to look at Treg products, for example, or some other kind of targets that might be impacting autoimmune disease patients. So we'll need to standardize the potency methods we use somehow. I'm not sure how we'll do that, but we'll need to work on that as an industry and see if we can come up with good solutions.
Maybe let's go down the line, Christine.
Sure. So we started the panel discussion talking about technological enhancements and how we engineer and manufacture these cells. And critical to their administration and data extrapolation are the technologies that are being developed to evaluate these cells, not only prior to administration, the ex vivo assays as we build the data packages and evaluate the autologous patient products or the allogeneic patient products, but that immunomonitoring that goes on. And so hand in hand, these advancements are really teaching us, right? I think critical from my point of view is you don't know what assays to build if you don't know what you're testing for. We learned a lot by looking at, how CD19 CARs mediate responses and toxicity, so CRS, neurotoxicity, other aspects, expansion. And so as we understand how these cellular living therapies behave and we advance these technologies for evaluating them, hopefully they'll go hand in hand and teach us and allow us to rapidly innovate.
Anything to add, Vicki?
Yeah, I think if I had 2 wishes for new tools, I'd hope that we could work together to develop a better in vivo model. It may be a bit contentious. Ultimately to inform safety in the human setting. So if we could have better models that would be predictive of CRS, ICANS, that would really help us accelerate then the dosing strategies and the clinical risks. And I think also I've heard Peter Marks talk about standardizing manufacturing for all of these cell therapies using similar delivery technologies. Whilst that's deplorable and we made the hurdle through the monoclonal antibodies, I think we don't have enough information yet for us to pool that knowledge together. And even if we did at this point in time, I think the challenges would be, can we still improve those therapies through manufacturing, through the different gene edits that we can look towards incorporating for those next generation therapies. So I think we haven't concluded the journey as yet to standardize manufacturing.
Yep, Michael?
Yeah, just really briefly, right? We're talking here about a strategy moving forward to have a real impact on how long it takes us and how much it costs us to evaluate iterative products, right? Products about which we understand a lot and we want to make them even better, right? And I think we talked about all the ways in which we're going to understand if what we're doing is riskier or better, but moving away from this, everything's high risk, we have to do everything for every product is really important for the field and important for us. So finding, understanding if it's a moderate or low risk and what that means in terms of product development is fundamental. And it also takes a partnership, right? It takes the academics or the biopharma, it takes the regulatory agency, and it also takes the institutional regulatory bodies, right? which in a previous panel was identified as something that we really have to focus on. Oftentimes, it's not the agency, it's not the biopharma or the academic, it's the institutional board that is concerned and makes everything high risk because you're safer if it's high risk. But that paradigm is, I think, part of what we're trying to change here today through informed discussion, data, and coming to the right conclusions. So.
And just to expand a little more on the idea of innovation, and specifically, I mean, there's many ways to innovate, obviously, but looking perhaps at the manufacturing innovation and how do we sort of bring more of the advanced manufacturing approaches into our processes and facilities. Some examples you could think of is automation, and you could sort of tag that with AI and machine learning and look for opportunities to mitigate some of the manual steps, some of the highly requiring training operations. So all towards the idea of how do we make our processes more efficient, less costly, and also more efficient. So just kind of want to wrap that up and leave those two thoughts about innovation and standardization as sort of something that could hopefully help take us to the next level and advance this field.
And I think we're getting booted off the stage. Thank you all so much. I think that was a wonderful panel. That's okay. Thank you.
Thank you all. Thanks very much. It was a great discussion. Really appreciate your insights and thinking about ways to standardize and classify risk, potentially down-classifying risks into the future for modifications of manufacturing these advanced therapies. We are now going to break for lunch. So for everyone that is joining the meeting virtually, we please ask you to stick around. We will be rejoining the broadcast at 1:10 Eastern Time for the remaining sessions of the day. And for those of you that are in the room, you should be able to pick up lunch outside in the foyer area that you came in through. And again, we'll reconvene at 1:10. So please enjoy. Thank you.
If everyone could find your seats, we're going to get started in about 1 minute. If everyone could please find their seats, we're going to get started in about 1 minute. Thank you.
One, two.
Everyone, please find your seats. We're going to resume the program now. Thank you.
Okay, thank you very much for keeping us on time and for everyone online for rejoining. I'd like to welcome our next panel up to go ahead and start grabbing seats on the stage here. The title of our next session is Opportunities for Innovative Trial Design and Regulatory Flexibility. We'll be building on some of the concepts that were laid out in the last session, as well as other concepts that were included in the white paper distributed for today. We have 2 more sessions left, so thank you for joining us. I'll turn things over now to our moderator for this session, Jonathan Jaziri, Executive Director of Global Regulatory Affairs at Kite Pharma, a Gilead company. So Jonathan, take it away. Thank you.
Thanks, Jeff. Ooh, all right, this thing is on and live. Good morning, good afternoon, everybody. My name is Jonathan Jaziri. I am the Clinical Regulatory Lead at Kite Pharma. I want to first apologize if I'm sounding a little nasally. I have an 8-month-old daughter at home. We just started daycare. And so for those that are familiar will know that this is an ongoing respiratory infection kind of situation. With that, though, I would like to first introduce our panelists here for the conversation today. First, I guess I'll start with Larissa Leptiva from the FDA. We also have Jay Park from Memorial Sloan Kettering, Raj Puri from Iovance, Nigel Yateman from Novartis, and Jennifer Yorling from representing Janssen. Before we get started today, I'd like to just set the stage a little bit and provide some context for the conversations that we've been having in the working group and as we've been developing the white paper. I think everybody here agrees and acknowledges that cell therapy is really the greatest opportunity we have to make huge advancements and transformational change for patients. However, the pace of the technology and the rate that it's advancing is faster than we can really develop and test these in the clinic. And this is really getting to the conversation from the previous session and Arie and Dr. Rosenberg's discussion around how they went from NK cells and IL to going to the CAR Ts and T cells. This type of iterative learning and training is currently happening right now by sponsors, but the majority of the sponsors that are doing this, we're facing the same challenge in that every time we make these iterative changes for these next generation products, when we go and have this conversation with the agency around testing these in the clinic, we're starting from square one. Not all next generation assets are created the same. Some will have greater differences and greater risks associated with it versus others. And currently we are experiencing that, yes, with each iterative change, we recognize there's a planned change to the therapeutic benefit. We recognize it'll be a new drug. We file a new IND for it. But the conversation with the agency is, okay, you file a new IND, you're back to square one. First in human dose escalation, 28-day staggering, sentinel patient dosing, 3+3 dose escalation designs, which really limits our ability to test these iterative changes in the clinic to really pick the winners and move them forward. If today we can walk away with an understanding that it's not just a black and white, there are gray areas in these new products, I think if we can walk away with that acknowledgement and walk away with a mechanism to have this dialogue between sponsors and the agency to discuss what these changes are and whether or not they are high risk, low risk, and what that really means, I think then we can walk away with a success today. So acknowledging that there's gray area, these aren't just all black and white, new products aren't all the same, and a mechanism for us to have this dialogue and engagement with the agency. So with that, I'll start with our first set of questions. So discussing, and to give some context around the types of products that we're discussing, the types of iterative changes, I'd like to hand it over and introduce Raj to speak about what he's been learning as the clinical learnings from these first generation cell therapy products, how they're influencing the next generation, what are these next generation products that you're currently working on and experiencing.
Thank you. Thank you very much. Before I start, I'd like to thank the Friends of Cancer Research for your kind invitation for me to participate in this very distinguished panel. And I'd like to, particularly, I'd like to thank Mark Stewart for organizing and working with us and in setting up this session. I must also say and congratulate the Friends of Cancer Research for coming up with a very impressive white paper that's very insightful, and I think I will encourage folks to look at this white paper that they have put it out. And I'd like to appreciate and give shout out to the FDA for the guidance document that they put out, a multi-version of cell and gene therapy product, an early phase clinical trial. I recall when I was at the FDA, I was involved in developing this guidance document. So I'm glad to see this out here and how the white paper concept is, has put in the context of that guidance document. So I'm going to talk about that in the learnings from the prior version of the product, and I'm going to talk about tumor infiltrating lymphocytes. And the pioneer is right here, my previous mentor, Dr. Steve Rosenberg, the inventor of TIL concept. And I recall in the surgery branch in the mid-'80s, I and Arie Belldegrun used to grow TILs together in the culture, in the same hood together. 38 years later, or 35 years later, 33 years at the FDA, and last 19 as a division director, here I am back to the circle and working on the TIL. I'm so proud that I work on the TIL therapy that for the solid cancer, which is, I think, it is the best hope for solid cancer, because that without a priori assumption of a target, here these TILs are polyclonal, and they will recognize thousands of neoantigens present on the target and attack them for the treatment of solid tumors. So the TIL therapy that we work for advanced melanoma, and we are pursuing that therapy, and we have learned that despite the logistical challenges and the complexity, such as surgical resection of the tumor, sending it to a centralized manufacturing site, keeping in track of chain of identity, chain of control, it is feasible to produce a product and scale and for a global treatment setting like we have done in our treatment trial. And so learning from that, now I see there is a opportunity to build upon that, on that TIL therapy as a technology, by evaluating genetically modified TILs. For example, by genomic editing technologies, one can knock down PD-1 or any other inhibitory molecules on the T cells, and we create next generation of T cell therapy. And perhaps these cells may go and survive better and do better in tumor microenvironment. And that kind of work that we are continually pursuing. In addition to, rather than getting a tumor from the bulk tumor, you can consider getting the tumor from the core biopsies. So there is an opportunity to enhance the learning from the first generation of the TIL cell products.
Excellent, thanks Raj. For I think it gives context for some different types of next generation enhancements. Nigel, do you want to speak a little bit about other examples for next generation cell therapy products to help give some bookend context?
Yeah, thank you. And thanks a lot for Friends of Cancer for organising this today. It's great to be here. Yeah, I mean, clearly we've learned a lot from the first generation products. And I think there's now, given the successes, certainly in the CAR-T space, there's a real sort of trust, I think, now coming in that therapeutic class, and that helps us going forward. Having said that about the successes, I think a big learning we have is that there's still a lot more to do. There's still a lot of ways we can improve. And if you look, for example, in third line large B-cell lymphoma, the PFS rate at 2 years is about 30–40%. So the majority of patients still aren't really getting long-term benefit there. So there is a lot of scope to still improve. And from a Novartis point of view, we're looking at what can we do to improve the persistence of the CAR T cells, and also can we get the product to patients quicker. So we do have now a new manufacturing process at Novartis called T-Charge, where the manufacturing is done in less than 2 days. And the idea of that is really to better preserve the stem-like memory T cells in the final product. So that means we're trying to get fitter T cells in the final product. And then we hope that then transfers into better persistence and then better duration of response. And also, obviously, with that short manufacturing, we're able to get the product back to patients more quickly than we've done before. And certainly in the US, what we're aiming for is to get that in under 10 days. So from pickup of cells at the clinic to then return of the final product. So we're really optimistic that this can help us build on the amazing results we've already seen from the first generation CAR-Ts.
Yeah, thanks a lot, Nigel. I think that gives the audience some context for the types of changes that we're talking about. You have those where are gene edited, and you're talking about new pharmacology, new things going on, whereas perhaps in the example that Nigel's given, you're saying, look, it's a similar construct to what we used before. We're making enhancements and improvements from a manufacturing perspective, recognizing there may be differences in the therapeutic effect, but again, these are two very different types of next-gen technologies, and right now, the lens is taking the same approach, at least from the regulatory perspective. So speaking a little bit from a regulatory perspective, the white paper, and I think as was referenced earlier, the FDA has issued a recent guidance document recommending a multi-arm or a multi-umbrella study, if you will, evaluating multiple similar related therapies. Larissa, can you speak a little bit from the FDA perspective around what the expectation is for sponsors to use this and how sponsors will be using this?
Yes, sure. So after all, this is FDA's guidance, and we published it in November of last year, and it pertains to situations when multiple versions or variants of a novel product can be tested in a shared clinical study protocol. And this is something that we believed would improve efficiency of the overall product development, because with one clinical study, we call it under the master's protocol framework, we call it umbrella study. So with that one clinical study and multiple product versions, you could have fewer patients enrolled, there could be some infrastructure shared. Actually, almost all of the study infrastructure could be shared. There could be a placebo arm that's shared among different study arms instead of having multiple studies done. So we believe this would be very beneficial early on in development of novel products, particularly when manufacturers would like to adjust the design of the product and not yet go into the full development because the design here could be optimized and adjusted based on some preliminary clinical data that could be obtained from that early study that's shared among multiple product versions. This is something that we believed would be helpful because just too many times we've seen these aha moments that would be occurring during product development, and sometimes they would be occurring in the later stages of product development when something would need to be rethought or redesigned, or when manufacturing could be changed. And on a number of occasions, we saw situations when product design could truly be improved at the point when the product already had almost all of the data collected for it, when the benefit was there, when the risk profile was already established, and at the time when the product goes on the market, there was no way to change the design. Yet you couldn't really risk mitigate by design. You had to either inform consumers about what potential adverse reactions to expect, or implement a strategy in the healthcare system. So really, this guidance and this ability to optimize product design early on is something that we believe is a great opportunity to improve the overall efficiency of development and to really save expenditures as well as the strategic development of novel treatments. Now, in terms of experience with this type of framework, sometimes we put out a new policy and we see a barrage of proposals coming in because people have been expecting this. This is not something that happened with this guidance. We're seeing some proposals and they're coming rather slowly, and people obviously understanding that this is IND related framework where one IND could be sent to the FDA, and that IND would be a fully fledged IND. It would contain all of the information about new product program and one version of the product, yet subsequent INDs, and that's the framework that's explained in the guidance, subsequent INDs don't have to be fully fledged. They only need to include information pertaining to specific product variants that are being tested. And with that, we start seeing more and more of these proposals, yet we're still not having enough experience to really make any systematic conclusions about what are the successes versus failures. We will probably see more in the future. But one successful, I guess, component of this is that recognizing this need for product design optimization early on, we were able, from the regulatory point of view, we were able to meet this need with adjustment of our administrative procedures and allowing manufacturers not to send in an IND, fully fledged IND every time, and just having developed this framework allowing to send only one large IND and being able to cross-reference with smaller INDs for the product variants.
Yeah, can I tell you from a sponsor perspective, we definitely don't enjoy sending the same information to the agency more than once for fear of getting different comments that we may not have gotten the first time, so I completely can appreciate that desire. I want to speak a little bit about the multiple products guidance. Not speaking from a kite perspective, but there was a publicly shared example from the NYESO TCR program from GSK that's now no longer being developed by GSK, but they presented the case publicly in the working group that we were, when it was kicked off and as we were developing the white paper. I'd speak about it a little bit because I was on that program back in the day, and I do think that it does exemplify a lot of what's in that guidance, and I think that the teams were able to recognize a lot of savings in terms of timelines, because if you have an existing protocol that's already running at certain centers and certain sites, and then you've got your Gen 2 and Gen 3, maybe you've got a TGF-beta version to help deal with tumor microenvironment, maybe you've got another version that's got some change in media to help with stemness. You don't know which of these technologies are going to work. And it may be that this technology works for this type of tumor type, and this technology works for that tumor type. So it may not be a one-size-fits-all or pick the winner and go, but we need a way to be able to test these in a single fashion, and I think that the GSK NYESO example of using this type of framework was able to leverage the tenets of this guidance, and I think they'd had experience, for instance, shortening timelines from from IND approval to first human dose, those types of critical timelines for programs. Jay, from your perspective, from a clinician perspective, I'd like to hear how this resonates with you and what sort of expectations you may see with this multiple versions guidance.
Sure. So, from my perspective in the clinicians and academic settings, and I think the efficiency, it's just cost effective and then the time saving too. So opening a single trial takes so much effort, not only the money financially, but the person and time, and then the time that requires from the initial review process. It typically takes about anywhere between 45 to 90 days to open a single study, often much longer, as you guys all experience from the industry academics. So, rather than opening 3 different studies for 3 different products for the same disease, having a one study and having an ability to add on as an amendment, which is kind of that itself is not too novel concept. It's just always amendment made, and we have done it in the past. But certainly it will be much, much efficient from the protocol perspective, academic and clinician perspective. From the patient-wide as well too, that you don't have to wait, and there's usually a null time between opening one study, closing that study, opening the second study, and then it's very hard to open the second study in all the centers when there's a competing study that's already existing. So that delays things as well. So having a single study with no other competition in mind and ability to add on a module or different product, whether it's combination, certainly increase efficiency. So I think experience-wise, we also try to do that in academics, and I think there's also a second example. The point is that I think that's where the close partnership with an academics, speaking from our experience, may be beneficial too, whether it's a one centers or 2 or 3 different centers, or whether you have an opportunity to test one product at one or the different centers, because it's the nature of being a kind of the smaller areas and then the nimbleness that if you want to test those hypotheses, early partnership will generate this data relatively quickly with hopefully a more kind of cost efficient setting as well too. So for example, one example I can think of at our center is that we had we are developing several different armor CAR T versions, kind of building on CD19 CAR T cells, adding on different cytokines or different armor to see whether those can improve kind of the further engage the immune system and improve the response rate and durability of the remissions. But we had 3 different versions, and we weren't quite sure which one would be successful. And then 3 were not ready at the same time, but 1 or 2 were, and then the third one was being developed. So in those situations, although there are other issues in the funding that we never got to do it, but we actually I wrote those studies, actually what we call Trident study, having 3 different arms, and it would be exactly kind of what the FDA guidance would have asked for to do them. So it would have been very nice design to do it, and it would be a smaller study. It does take more time at the beginning kind of planning to do it, and then, but I think in the end it will come out and better. We'll actually save more time to truly see kind of one product. And to your point though, it may be for different disease, one product may be better than the others, but at least we'll have an opportunity to test in a kind of single study and a good translational study, and then the real time and very close collaboration and monitoring the disease outcome actually will be critical for this too, because you don't want to wait until several more patients are treated. So you have to actually constantly have to evaluate kind of what are the outcomes and safety and efficacy of these products to make a relatively close decisions and early decisions of whether to move forward or not.
Yeah, I think it definitely helps from a data evaluation perspective. And I think the point that you also made there is key for everyone to appreciate that these next generation programs aren't available necessarily all at the same time. Sometimes they're in close succession, but sometimes there's a bit of a lag time between. And having an existing opened study protocol, contracts that sites are already approved, being able to have all that infrastructure set up really helps that 3rd, 4th iteration kind of come through.
Just one quick add is that I think we had an original in the 1920s, the CAR T cells in ALL. We actually went through the many versions because, as Arie was kind of alluding to earlier, the conditioning chemotherapy, we weren't quite sure what's the dosing. The very first cohort did not have any conditioning at all. So we then added an amendment and to do it in a single study rather than opening another study to test the same concept. So we were able to do the relatively real time making those changes and the different dosing based on disease burden. So in a single study that allow us to, rather than closing and opening and closing and opening in a single study to do it, it would be much quicker. We learned a lot through the process with relatively few patients.
That's great. Thanks for that insight. So a lot of concepts in the guidance the white paper talks about accelerating clinical development, but as several of us know, the rate limiter oftentimes isn't the clinical development, it's the CMC side of it and the quality side of it. So Jennifer, what are some thoughts you have around how we may be able to accelerate the CMC timelines, or what are some implications as we accelerate clinical for CMC to catch up?
Yeah, it's a really good question, and I'm really glad that you brought it up because I do think we get excited about the opportunity for data extrapolation in the clinical setting to leverage your experience to establish your starting dose or maybe reduce the staggering that you may need to have or define your DLT criteria, and these were all discussed in the first panel. But I do think data extrapolation could also be leveraged in the CMC space. I mean, the white paper does an excellent job, like Dr. Puri mentioned, that to identify some opportunities that we could be leveraging within the CMC space to extrapolate data from a primary product that we've established the experiences with to apply it to the secondary product. So things like stability or your analytical methods or reducing the amount of engineering runs you may need to establish your IND. I mean, there are a lot of opportunities that laid out in the white paper that I think our sponsors should be considering as we accelerate the CMC components of drug development for CAR-Ts. I'm also really encouraged to see all of the movement that we've seen at the FDA with regards to helping us identify opportunities for cross-referencing, to eliminate the need for the administrative aspects of representing data, as well as the platform technologies pathway that's coming from the legislative bodies to help us identify opportunities for establishing precedents and leveraging existing data in the CMC space. I think that also the CMC readiness program, the pilot program that the FDA has just announced, I think will have a lot of opportunities for us to learn from those experiences. We look forward to the FDA sharing the experiences with us through either guidance documents or Q&As or these types of forums to leverage the knowledge that are gained through those pilots. So I do see a lot of opportunity to get the CMC development to be lockstep with our clinical development, hopefully reduce that disconnect between clinical and CMC timelines to get these patients the products that are so promising to their armamentarium.
Yeah, thanks, Jennifer. Appreciate that. Yeah, it's something that we face. I think everybody faces on a day-to-day is trying to address CMC challenges, manufacturing challenges. It's a process, not a product, and it can be, as was mentioned earlier, the two go hand in hand for this type of modality more so than in conventional therapies. Raj, from your perspective, what sort of ways can we help accelerate the data extrapolation from the quality side?
Yeah, so I think that to add on to Jennifer's good comments, referring again to the white paper as well, that one can consider, because the related product, the next generation related product, requires good assays, which is a release spec assays, for example, potency assay, et cetera. And all of these assays has to be qualified and later on validated. So perhaps one can consider a platform approach where the assay is qualified for product A, and the same qualification applies to the product B, saving time and resources. And as you determine which product is going forward, A, B, or C, taking this qualification data that you already have done for one, extrapolate it to the second, and as the product move forward, then you validate it with the validation before embarking a pivotal trial, and using and of course identifying the range of release spec based on the clinical experience. So I could see there is an opportunity here for the release spec of the qualification. Similarly, a platform approach, sort of like a potency assay. If the mechanism of action of related product seem to have the similarity in that, so when you come up with either potency assay by itself or the matrix approach that the FDA guidance had put out it's a 10-year-old guidance document which perhaps need an updating, but the concept is there. If you have a matrix approach that could be qualified for product A, that same principle can apply to the product B. You don't have to repeat the whole thing again. And then when you advance to the next phase, so the pivotal studies, you just validate for one, not for all of them. So I think that's an opportunity here to saving time and resources.
Yeah, I like that sort of mapping where it's the consistency across, and I think a lot of sponsors will appreciate that, especially there's a lot of iterative programs out there that, for potency assay, for instance, no need to recreate the wheel if it's already working the same target. So changing gears a little bit to clinical trial design considerations. Nigel, over to you. What are some trial design considerations for next generation cell therapy products, particularly when the first generation cell therapy product may already be approved or moving into earlier lines?
Yeah, it's a fantastic question that it requires a lot of debate, and it's great to, again, to have this forum to be able to do that. So I think what that's leading to, the implication of coming into earlier lines, is gradually we're going to see standard of care becoming CAR T therapy in certain indications, and we already see that in second-line lymphoma relapse within 12 months. So with that situation, a developer with a next-generation product, if you took a traditional clinical development plan thinking, to develop that new product, you're probably going to need to do a trial using the standard of care, which in this case would be then the CAR T. So on paper, that sounds like the way to go, you know, as a development plan. But I think probably most people in this room can imagine that's a really tough challenge to do, and in practice, it's probably just infeasible. And I think really there's two main reasons for that. First of all, to do such a trial, it would require a lot of patience and a long time to read out. The reason for that is because, yes, we've seen some great results with CAR Ts in randomized trials to date with hazard ratios less than 0.4. It's amazing results, but that's against a comparator that's not a CAR T. And I think if you're in a situation where you're going to compare against an existing CAR T, that would be really optimistic to think you could see such a big treatment effect. So what you might want to do is power the study for a narrower treatment effect, but that necessarily then means more patients. And some of the things we've looked at it, is maybe a more realistic hazard ratio is 0.75. And just to put that into context, that would be like, for example, moving from a median PFS of say 18 months to 24 months. So something that is meaningful, and especially if there's other benefits, you know, with the new product. And with that sort of hazard ratio, the trial, you would need over 500 events to be observed. So you're looking at, I don't know, 800-900 patients, and probably taking 5, 6, 7 years to read out. So that's a huge undertaking, obviously, for any single manufacturer to do. And even you could say in this space where we are, where there's so much innovation and so quick innovation, you even wonder by the time you get to the end of that study, the result might even be less relevant because the changes that have already come elsewhere, you know, in that time.
Landscape changes and such come along that time. Mm-hmm. Yeah, I think one of the other points that we were discussing was like how do you go up against products that have curative intent, right? When CAR Ts are moving into earlier and earlier lines, and it's been established that CAR Ts have this curative intent treatment, the traditional endpoints that we've been looking at, survival and PFS, are these the appropriate endpoints that we should be considering, or is this an opportunity for new novel endpoints, given the kinetics of response and the kinetics of the drug that we're working with here? These aren't the traditional modalities. We should be thinking maybe differently around clinical trial design and endpoints.
Yeah, no, sure. And I think, I just want to finish what I was going to say.
Sure,
actually,
sure.
yeah, no, sorry. So one thing is having a large study, and that's an impediment. But a second point about that sort of trial is, because we're talking about CAR T therapy, if you did that trial, even if you took on the fact that it's going to be long and everything to finish, the manufacturing obviously is part of the product, and the manufacturing is done on the control arm by potentially a different manufacturer, a different company, and that's done in real time during the study. And of course that's tough logistically to organize, but also the manufacturing on that control arm is going to be done like in the commercial setting, if you like. So with the vagaries of sometimes delays or whatever, and put that on the other hand with the experimental arm. So the sponsor is running the study with their new CAR T. They're completely in control of the manufacturing. They can plan for capacity to support that trial. And so very likely what's going to happen is you're going to get much better turnaround times on the new product compared with what you've seen in the control arm. And so, and we know that obviously earlier shorter turnaround times are generally going to lead to better outcomes. So you could do that study, get to the end, and then even then the results maybe are going to be difficult to interpret because there's this confounding by potential bias coming in.
Your
because
standard of care may be underperforming, is what
yeah,
you're getting at. Yeah.
And in fact, it's favouring the new CAR-T because it's going to make, but that, you know, so it's a huge investment to then maybe even end up. And I think if you compare it to other, like for example, checkpoint inhibitors, you can really compare those and expect to be able to get a good result at the end that's interpretable. But here, because the manufacturing is such a key part of the product, I think that's really going to be difficult to do. So
it's kind of like what you're alluding to is maybe more use of real-world evidence and that type of an approach.
That's one thing. I mean, you know, we've been thinking about this in Novartis in the biostats group about, and I'm a biostatistician, so yeah, and where to go with this. And yes, I mean, one option would be a single-arm trial, and you contextualize it with real-world data. I think, realistically, that isn't going to be acceptable from a regulatory viewpoint. So we'll see, but I think it's tough to think that that's going to work. So
I see Larissa as waiting to comment on that one.
Yes, yes, and of course, listening through all of the challenges with how to conduct trial, what is the best population to choose, how to move product that started maybe as a last line therapy a bit forward to try to use it in a different patient population with different prognostic factors, right? So it's not entirely the same thing as to when we're looking at testing a product in a relapse refractory disease versus as first-line therapy, where there are already products available on the market. And in order to really offer advantage to patients, you would like to show somehow, right, to demonstrate that the product is as efficacious or maybe even more advantageous, more beneficial as the first-line therapy. So you obviously have to take into consideration the overall landscape of the treatments that are available for the specific patient population that you're, you know, aiming to address and to target. But I also would like to mention a program that we have currently at FDA, and that is called Complex Innovative Design Program. This is specifically talking about newer statistical designs for trials that are intended to demonstrate the primary substantial evidence of effectiveness. And this is the program that was started as a pilot with the last reauthorization of the Prescription Drug User Fee Act in 2017. And now it is a fully fledged program. And in the heart of that complex innovative design is the use of scientific computing. And that's when you could do computer simulations to determine the trial operating characteristics, type 1 error and power. That type of design also gives advantage because it uses borrowing methodologies from the Bayesian statistical methods, and therefore, again, fewer patients, less resources perhaps to be used than the traditional frequentist-based statistical approach type of studies. So we have actually some information about this on our website, and there are examples there because typically people who are, well, manufacturers, sponsors of new products who are enrolled in that program would agree to disclose some of the main features of the study design that's been discussed for the field to see. And so we have some examples there. For example, there is a master protocol in pain where they were looking at different treatment options, again, choosing the most optimal treatment option. And then there is an example of a protocol in diffuse large B-cell lymphoma when a commensurate prior was used with a specific model to borrow information from external controls. So all of these various types of designs is something that we consider under this program, and we grant additional meetings with the FDA to discuss these types of design. And maybe this is something to consider when looking at next generation of cellular therapies, particularly in settings when there is prior knowledge about a product or about the patient population that you're working with.
Thanks, Larissa, for the plug for the CIDP. I would agree. I think it's a good place to have these types of conversations with the agency. Jay, from your perspective, from a clinical perspective, what type of trial design considerations should we be thinking about for these types of next generation products?
Yes, I mean, I think ultimate goal here is, and there's no one size fits all, and I was kind of sitting here thinking about the ultimate goal is to minimize giving ineffective or less than optimal therapy to patients and deliver more effective therapy to more patients. So that's the whole point of kind of us sitting here increasing the efficiency and then it's hopefully eventually to more effective drug development there. So I think to Larissa's point, and the one thing is that new innovative design, I think is a cell therapy. I think this has also been done in targeted therapies and many other phase 1 clinical trials, similar ones. But I think we're also beginning to explore, and I think there is kind of the benefit for some of those things, and whether there's prior knowledge there, and even when there may not be prior knowledge, and I think because I think what we have seen in the cell therapy also is that the cell dose question is the highest dose may not be the most effective dose or most optimal dose for these patients too. I think which is a traditional drug development in cell therapy, you may be able to achieve that in the middle dose or the lowest dose, and there's a fair balance of the safety and efficacy with all the drug development there is. And we've seen that in kind of in cell therapy clinical design. So we've been also beginning to explore, I personally been interested in beginning to explore innovative study design. So we have adopted a couple new cell therapy studies, and for the first time I'm looking at the kind of Boeing study design or CRM or continuous response monitoring to better assess that rather than doing 3+7, 3+3 and increasing, but kind of more real-time monitoring of the safety and efficacy so that you can expand the cohort where you think you're going to see the most efficacy earlier on rather than waiting until the final dose and you do all 4 dose escalations and then you go back and it's okay, let's expand dose level 1 and dose level 2, 3 or something like that. So I think exploring this innovative study design is one way I think that we can hopefully increase the efficiency and trying to find the optimal dosing earlier on and save ineffective dosing or unsafe dosing to less patients. And then I think it's a more difficult question, as what also Nigel was also alluding to, is as the therapy is kind of moving forward, what is the optimal study design? And I don't have a great answer to that one, but I do think that if this, the modification is relatively small, and this is where there's a lot of variance in definitions of what are small changes, and at what point it becomes a new product versus the small improvement of the kind of the older product. Because we don't want to keep giving, if we have a second generation, third generation, like different software, for example, even though it's not really the exact proper analogy, that you don't want to kind of stuck with the first generation because that's kind of what's been approved and that's kind of what you can use the most. You want to use the kind of the next generations. And with drug development like this, when things are happening relatively fast and deliver them early. So I think we have to agree on, and there are some criteria as to what is going to be considered, how much of a change will be considered a new product, and that will really depend on how much it will change the quality of the product in itself too. But if the changes are minor, then you don't want to start from the very beginning, kind of the drug development dosing too. If you already have effective dosing from the prior one, and it's a minor change, at least that provides a ground of where you can build upon, so that you don't have to start from the very beginning. And then that will hopefully allow us to kind of move this therapy to kind of earlier lines of or kind of earlier drug development there.
Yeah, no, thanks a lot for those insights.
Can I just back to Larissa, so that complex design initiative in FDA, I think it's great to have that for sure. But I just wonder in our field whether there's enough commonality that we can merit, like ourselves, trying to find some ways that can help across a broad range, rather, because that's really for a specific protocol to go to for that initiative. And yeah, I don't know. I mean, it'd be great to think if another white paper could be considering how do we push this forward? Are there some ways, general ways in CAR-T when you want to go to a next generation?
Innovative trial designs for cell therapy is kind of a white paper. Yeah. That may be a future plug for those in the friends that might be thinking of another initiative to be working on. I do want to just mention we have a little bit more time left. We will be taking questions from the audience, both here and virtually. If you have questions, I did get a couple, so please feel free to continue sending those through if you have some. I do want to move to this next question, though. The white paper lays out an assessment aid-like document, which leverages or describes how sponsors can leverage this tool to facilitate the internal discussions as well as external discussions with the agency. Jennifer, from your perspective, how do you think sponsors could use such a tool or document to help in those negotiations?
Yeah, I thought that the assessment-like aid is going to be very helpful for this space, especially for data extrapolation. Now, I know that the concept of cross-referencing is not a new concept for those of us who are in the drug development space, and it's already been recognized on this panel with the primary, secondary IND and the opportunity for umbrella trials, et cetera. But I think the assessment aid is a really nice tool for us as industry to consider driving internal conversations about opportunities for leveraging existing data from a primary product, like was being discussed by Jay, and earlier in the first panel, is like you need to, if you have enough information from your primary product and you want to leverage that information for a secondary product, leveraging this assessment aid to be able to have that conversation both internally and with the FDA, I think will be quite helpful in driving this innovative and incremental or iterative development paradigm that we've been discussing today. So I can imagine that, or if you've seen in the white paper, there's this appendix that gives you an outline of a template that could be used for the driver of this discussion, where you explain what we understand of the primary product, what we understand about the secondary product, and outline what those risks are with the data gap that you have between those the differences between the two versions of products, and then what that data generation strategy would be to address those risks that are present between the two products. So I think this assessment aid not only could be used internally for driving that strategy, what would your IND need to look like, it also could drive a pre-IND conversation with the FDA and ultimately become almost like a reviewer's guide for the FDA when they're getting a secondary IND to understand what is being leveraged from the primary IND towards the secondary IND and hopefully enable efficiency of that review process when you're submitting that IND for the secondary product. So I think it's a great tool. I hope everybody uses it, and I hope that the FDA may even consider piloting this process with their in future experiences, maybe even through friends, like another opportunity for generating more experience.
It'd be great to learn maybe over some time after the agency's had some experience with this, what their lessons are. Larissa, from your perspective, what's your thought or the agency's thoughts on such a assessment aid-like document?
Well, in general, we've used assessment aids before, so we've used them for other categories of submissions. We've used them for BLAs. Assessment aids are helpful because they help to orient our review staff in the proposal, in the submission, understanding the rationale for what's being proposed. I'm pretty sure, and what we've heard from Jennifer, assessment aids are also helpful to industry to organize their thoughts, to crystallize their strategies for what they will put in that submission. I saw that assessment aid as a helpful tool. Right now, it's probably still more on the at the conceptual stage, on the conceptual level. Obviously, as was alluded here before, different products will have completely different strategies, and it would be probably some time from now, we will have an assessment, hopefully we will have an assessment aid oriented to, say, CAR T therapies, and there could be an assessment aid oriented to T-cell receptor therapies, or an assessment aid oriented to another category of products where there would be a much better understanding of where that strategy of data leveraging and cross-referencing should go for a specific product category. And it would also be helpful for us to know when these proposals are sent to us, what is the impetus for this next generation product? Because we really want to bring more benefits to patients, right? We're there not to promote the second mover's advantage. We're trying to protect the interests of the consumers and make sure that the next generation product is somehow beneficial to patients. Is it improving efficacy? Is it allowing to see more durability of the response? Does it lead to fewer or maybe less severe side effects? Is it more convenient in administration? Maybe less invasive? Or does it improve the manufacturing so much that the costs have would be decreased so significantly that the costs then would not be passed on the consumers in the form of the very high prices for cell therapies and gene therapies, as we all know very well. And so in each assessment aid, it would be helpful for us to know what is the driving factor behind the new generation or this next product, next cellular therapy. And of course, the general proposal categorically as to which data could be leveraged, which data could be cross-referenced, and where we need to generate additional data perhaps to demonstrate that the product is as good or better.
So I think having some understanding of what the limitation of the first generation that you're trying to solve for, I think that may be a good starting point for some of these assessment aids.
That's true.
Right? Yeah.
Absolutely.
Don't be shy of explaining what are
Yes.
the downsides of your first generation.
Yes.
That's how you're going to make the second generation shine bright. We do have another question here, but I want to take a question from the audience, the virtual audience. This is really for the whole panel. With such a large pipeline that will move forward into the clinic and participation rates that's still estimated to be around 5 to 10% of the eligible population, how can we work together to improve trial awareness and recruitment? This is something that I think faces everybody. Jay, maybe from your perspective?
Sure. Difficult question. I think it's true for all type of studies as well, but I think the cell therapy, some of the challenges, and I think is not only the clinical trial, even if the approved therapy, not everybody is traveling to kind of receive this approved therapy, even though it is effective and better than the chemotherapy or other standard of care. I think there are many areas that we have to kind of work on improving the safety and increasing the access, which was also brought in the first part of the talk as well too. Because currently this CAR T-cell therapy or the cellular therapies are being administered in a very specialized or the transplant or cell therapy centers, which is not all infiltrated closely to every living areas or especially the rural areas. So this is kind of concentrated in more larger setting or metropolitan areas. So people have to make an effort, the doctors, the community doctors, where majority of the cancer patients, including these heme malignancy leukemia patients, in the vast majority are still being taken care of by closer to their home community oncologists, and there's nothing necessarily wrong with them and that they want to stay home necessarily, but they'd also make it very harder to cross the border or to cross the bridge to across another city and a center to receive this therapy. So I think we trying to, there's no perfect sense, but we try to first increase education to the community, the local oncologists to make sure this therapy is available and there are help that we can actually deliver better, meaning the housing effort or the transportations and other things. So there are a lot of help from the industry and then also the philanthropic effort from the institutions, trying to make the patient experience as seamless as possible, less disruptive to patient lives there too. The second, I think we have to improve the safety of these products, and then certainly this requirement for the 28-day of training, which is put in place because of the side effects of a certain cellular product, was mainly CRS and ICANS. Because of that, those things can happen in the early phase of their lives, the treatment, and that they have to maintain or remain close to their centers within 1 or 2 hour living distance, which also put a limitation of the whether, especially for the cell therapy, to do it. Now, as we are getting better with the safety product being improved, safety of such products being improved, I think that limitations, that regulations need to be re-examined and to be shortened. Maybe not all cell product is the same, and therefore not all cell product requires 28 days kind of the viewing. And we have seen some of the solid tumors and TILs, and most toxicities happen in the first 7 days. Why do they have to be in the hospital for 10 days or 28 days nearby, that they all recover their normal blood counts and so forth? So I do think that there are, as we get more cell product in those are more approval settings, that we have to kind of look at it more closely, that these limitations that we put on for safety, which is appropriate, but as we get more experience with those need to be kind of the shorter and shorter, or ensure the safety of the patients, but within reasons to do so. The clinical trials, I think, argument that I think it kind of goes a long way. That paints a picture of a clinical trial for cell therapy, so patients may not be traveling. So I think we as an oncologist and the academic clinicians, and then I think the community can do a better job of advocating, yes, these are effective therapies. They are help that there are means that we can help patients get through. And then most of the cell therapy is really just at the beginning, kind of in the first month. If you put in that extra effort in the first time, then you may potentially be able to get months and hopefully the years of a durability remission, rather than getting to the infusion centers every week or every month to get the monoclonal antibodies or the chemotherapy. So that point, I don't think is well made. And in order to do that, we do need to develop the durable remission therapies. And having all the collection and others, it's kind of the issue there to the autologous products. So having more of the, hopefully the off-the-shelf product will increase its access as well, because getting the leukapheresis center to get the cells collected, there's a lot of a bridging time as well too. So not everybody can get to the first step to collect the cells and then make it to the cell infusion. So there are at least two steps kind of before getting in. So the idea of even traveling, and then the disease has to be well enough to do it. So there are limitations which we're trying to overcome. So having a more immediately available therapy, hopefully less toxic, will bridge that gap, and the more patients can access to it. So I think all of the things that we're doing, and then some of you guys are kind of doing and developing these therapies, hopefully will lead to the more participation in such clinical studies, and more patients receiving such products.
Thanks a lot for that. You actually answered one of the other questions around that we received about whether or not there'll ever be an opportunity to reduce the number of staggering less than 28 days, and you kind of discussed that will bring us, bring better, safer drugs, or drugs that have a different safety profile, and then reevaluate the DLT window. I do see that there's a question here in the audience that I'll take before we get to other questions.
Thank you. This is Jiawen Zhu, biostatistician from Genentech, with a research background in novel clinical design. And I actually work in one of the CID quoted by the panel today. And so my question is about different versions of those products. And sometimes the second version remind me about like those new version of device. So my question is on like some of the learning on the device development, on study design, and some of the method like leveraging Bayesian method, maybe can be leveraged in thinking about those novel cell therapies. So I wonder whether there is some past discussion on how those may apply or may not apply to this novel topic.
So maybe the question, Larissa, from an agency perspective, is there cross-learning? That's what I'm gathering from this, cross-learnings and trainings across the centers.
Right, so the question was about what we have learned from the device industry. Oh, well, from device industry to cell therapy industry, right? So that's a bit of apples and oranges. I'll say that we're still learning and cross-pollinating each other. To see where the right, potentially applicable to all methodologies and strategies could be. I can't really speak a lot about the device industry. I come from the biologics and drug side.
Thank you. Thanks, Larissa. I have about 40 seconds left. I'm going to try to shoot one more question in there. So I think this is an interesting one. For products with multiple engineering changes to improve benefit risk, will the FDA be looking for contribution of individual engineering components? Should each individual engineering component be tested through this umbrella master protocol design, or will there be flexibility perhaps to combine them into a final product? Larissa, I'll start with you, but I promise I won't just leave it to you.
So which flexibilities, right?
Yeah. Is it expectation essentially that if we have multiple changes we want to make, can I stack them up and put them into one, or am I going to be expected to test each individual one separately, demonstrate the contribution of components of each change before I put them all and stack them on top of one? What's the expectation from the agency?
So obviously, we're in the business of regulating individual drug products, right, or biological products. Oftentimes, they're used as therapeutic regimens, and this is a known knowledge, right? And when a specific drug product or biological product is claiming to be effective and safe, that claim of efficacy and safety has to come specifically in relation to that drug or biological product. Right now, the question comes when they're used in regimens, and when maybe in that regimen there could be, say, an approved drug product and an investigational drug product, and so the devil would be in the details. But the main concept here is that if we're talking about a specific therapy, that therapy, when it's claiming to be effective in a particular type of cancer, should be demonstrated to be effective in that type of cancer, right? And then things could be put into regimen.
Thank you. I have the blinking red light, so I think we'll move to the next session. Thank you, everybody.
Thank you very much, Larissa. With all those softball questions, I hate to cut things off. But we are going to move to our final session of today. I'd like to thank the previous panel for all of your thoughts and help in constructing the white paper. I think we heard some great ideas that were reiterated there in terms of trial design, use of umbrella trials, the potential for an assessment aid to improve communications. So let me go ahead and welcome the final panel up to the stage, if you don't mind making your way up. We'll get things situated for you here. And I'll start by introducing our moderator, who is Margaret Anderson, Managing Director from Deloitte and also a board member at Friends of Cancer Research. So I'll invite our panel to come on up.
Hi, everybody. All right, so I think I'm all the way back here.
Sir?
Okay, no, not working. Okay, apparently this is working. Yes, it is. Excellent. Wake up, everybody. So we have a terrific audience here in the room, and I know there's still many people who are watching the webcast. So this has been, I think, a very, very fruitful conversation, a very complex conversation thus far. So in this panel, we're going to do things a little bit differently. Instead of taking a siloed approach of each of you, sort of giving a presentation, I want you to crosstalk. And Arie said maybe cross-disagree. So he was interested in there being some, I don't know, contentiousness or just, some liveliness. So I did want to start, though, if you look at the title of the whole meeting, it was about creating a blueprint. And so as I was listening, I just Googled the dictionary definition of blueprint, and I want to read that to you. It's a photographic print in white or blue on white, used for copying maps, mechanical drawings, and architect's plans. Now, Ellen, I know that you and Jerry Siegel have a long background in construction and building. Maybe come up to the mic for a quick second. I know, I told her I was going to put her to work. Ellen, I wonder what the utility of blueprints were in your work when you were doing construction. If you could share
Don't
why were they
forget,
important?
I was the developer and Jerry was the builder, and my job was to yell at him that the plans were not right. So the architect developed the plans, and the builder seriously had to follow those very specific plans. But I can tell you that never really happened, and all I can tell you is they were very, very heavy. So and they were multiple pages, maybe 100 pages, and to lift one set of plans, that there was the mechanical and the electrical and all of these other things. And even in those days and today, still not a whole lot of coordination. I had a building that the elevators were placed in the wrong place and everybody blamed one another, and that was on the blueprints. So that's giving you the complexity of what we were dealing with, and still will, and start about science is a whole other deal.
So the reason I wanted to focus in on this word blueprint for this panel with this august group represented here is to kind of start with posing the question around this area, this field. Is it possible to create such a blueprint? Because listening to the conversations thus far and, you know, kind of the history of the science, it's moving at such a pace. I don't know how possible it would be to replicate such a blueprint. Peter, I'm going to start with you. Talk about the role of blueprints in the regulatory paradigm and then relate it to this.
Well, there are blueprints like developers use to build buildings, and then there are blueprints like I use to build a doghouse. So there are various types of blueprints. So we may be able to get to a blueprint that's somewhere in between those two. It may not be at the extreme of, it's true, I mean, I know from when we moved on to the FDA campus, we had blueprints and they filled a room. We may not get to that level. On the other hand, it's probably not going to be like the sketch that I have of how I'm going to build a doghouse. So there's going to be something in between. And I'm probably going to be in the doghouse shortly. The issue is, I think we shouldn't let perfect be the enemy of good, and we probably can find some things that help us find ways to expedite the development of these products that streamline things. And I think everything we can do that streamlines the process of development more can help get products to patients more quickly and hopefully at a lower cost, which will also help us out here.
So, Peter, just to drill down on that, so when you were building your doghouse, you wanted it to be functional, maybe the dog needed to be outside and have a little shelter, whatever the purpose was. And when you're making these decisions and you're charting the course at FDA, you're trying for the same thing. You're trying to be functional, efficient, make it structurally sound, but not take 10 years, since sadly a dog doesn't live that much longer. So how are you all thinking about your blueprint for this space, given the pace of science? Because that's the thing, I mean, on the one hand, we could say the pace is pretty rapid. On the other hand, listening to what you were talking about at the Fireside chat, we've been thinking about some of these areas for a while. But how is FDA positioning towards, sort of what we're starting to see is more exponential pace?
Yeah, well, this is a problem in cell therapies, I think, because you have something that's going to be constantly in evolution in a way that's very different than small molecule drugs, right? I mean, once you decide that you've got the structure of a small molecule drug, yeah, you might tweak the synthesis a little bit this way or that way, and it's true that in some cases there are natural molecules that have subsequently been synthesized synthetically, and that's been a big breakthrough. But it's not like the difference of what's going on in cellular therapies now, where I think we're seeing generations where, because of our understanding of molecular biology, because of our understanding of what we can do to tweak these products, we're going to be wanting to see generations move every several years forward, and that's going to present challenges. And I don't think we have all the details worked out on fixing all of those things yet, right? Because I think there's the issue of how do you make what changes are acceptable before you have a new product, right? And I think we're trying to help define that and understand. I mean, yes, we have a definition, but the question is, where should that boundary be, and what could we do to allow us to bootstrap from one product to the next without having to go through another full series of clinical trials? And again, I don't know that we have all the answers, but I think we're going to have to find our way through them.
Absolutely. So, Arie, let me ask the same question to you. What do you think about when you think of this, building this blueprint out? I mean, you've been working on a blueprint your entire kind of career and life's work. Where do you think we are today in terms of the construction of that blueprint for cellular and gene therapies?
Well, I think it's a moving target. When I'm looking at a blueprint, I can understand Ellen and her construction company doing a blueprint. But I'm trying to think back several years ago when the FDA came for the first time to inspect the first manufacturing plant in the country, in Los Angeles, and that's your old kite, you know, our old kite, your new kite. And at that time, they came for 2 weeks. And we said, what are they going to ask? Where is the blueprint? I can tell you, and Mark Better was through that, and David Chang, and a whole group of here, and Zach, they all been through that. The FDA had no question, there was no blueprint. I wouldn't say the blind led the blind, but we all came together, working together as a group, and after 2 weeks, it was a coherent group that said, you know what, we need to continue to improve. And we took a full picture of 20 people, all of us, the FDA, I've never taken a picture with an FDA guy, but you know, we have a picture with FDA because we all felt that we are creating something together which will change the industry. And unfortunately, it was lost a little bit in translation these days, and we need to bring it back.
So if we think of this meeting as part of that process, I'll go to you next. What would you put into the blueprint process? I'll start with you. Yeah.
Me? Yeah. Oh. I was going to say we don't need a blueprint, but okay.
Please, all right, throw the blueprint out.
Look, we're not here to make blueprints, just like I used to tell my team that the goal is not to make a PowerPoint presentation. The goal is to get better drugs to patients. And at the end of the day, the blueprint is it's a way to make sure that we all are on the same page when we communicate about what we need to achieve. You know, when COVID hit, there was no blueprint, and yet we got vaccine out in record time. And that was because we got together to figure out what needs to happen, and we made sure that we iterated on it in a very rapid pace to meet the needs. So I think for me, the relevant part here is actually not having a blueprint, but having the conversation around the things that matter. And the blueprint comes after that to make sure that we're all on the same page, and it enables those that are not as part of the conversation to still benefit from the conversation. And again, that example for me was very seminal because the impetus of time meant that we had to get a phase 3 protocol at the same time that the FDA had to put a guidance out together. And we were the first out of the gate, Moderna at the time. Our phase 3 came out the same week that the FDA guidance came out, and it wasn't luck that the two were matched up, right? It was communication, and that allowed every other company then to have the same guidance and be able to execute to the same phase 3. And I think that's sort of what the goal is here. It is to make sure that we are talking about the right stuff so that we all end up on the same page.
So Mark, representing the Melanoma Research Alliance, which is an entity that focuses on the scientific underpinnings for melanoma science and advances, but is very closely tied and connected to patients. Talk about the patient perspective in whether it's a blueprint, an anti-blueprint, collaboration, communication. You know, how do we make sure that the patient voice gets inserted into this dialogue?
Yeah, no, a great question. I think, well, first, we all don't want the elevator in the wrong place. So I like the blueprint analogy, but I think there can be a blueprint, you know, across solid tumors. So MRA worked with Friends of Cancer and other groups to look at commonalities in defining PD-1 resistance across melanoma, lung, and kidney. So I think there's some commonalities that could lead to a blueprint. I think from melanoma patients' perspective, it's really complicated and complex very quickly, as you know, as a former CEO. You know, there's lots of treatments, you know, 3 different approved immune checkpoints. We're very excited about sort of off-the-shelf cellular therapies, hopefully coming soon. And then I think it gets really complicated when you think of some of the mRNA vaccines, which there's some that are being developed that are more personalized and customized, some that are more off the shelf. So I think it's an exciting time, but it's also very complicated. And then the last sort of, well, not the last, but another dimension I would add is this whole idea of the role of the microbiome. And you know, clearly when it comes to FMT transplants, you know, there's clearly signals. So how does this all integrate together? Is it what stage of patient? Is it treatment resistant, treatment naive? Where do we integrate these new things? So I think there could be some common blueprints across solid tumors, but I think melanoma patients, especially those that aren't responding to treatments, are really eager for new options.
So bringing that patient perspective forward into this conversation, if patients really are the ultimate reason that all of this is happening, I sat there today listening and I thought, wow, this sounds really exciting. I'm not a scientist. I've just worked in the area of my whole career. And I thought, if a patient sat in this room, they might think that, we are literally on the doorstep of fixing everything, which I think all of us in this room know not that easy. And in a way, COVID, as long as it was miraculous, right? I don't think people truly understand some of the underpinnings of, what existed, what it took to get us, over the finish line in terms of approved vaccines. But let's think about it from that patient perspective of, hey, I have a diagnosis today. So what is standing in the way of, sort of advancing this? Is it, if there are things standing in the way, is it science? Is it money? Operating models, regulatory paradigms, more meetings like this so you can sit and talk? Anyone who wants to start the dialogue, go for it.
Yeah, let me go for it because melanoma is now close to my heart. Look, first of all, it's the science. I mean, let's start there. We're fortunate to have this conversation because we have effective cell therapies. And the cell therapies work. In fact, they work so good, as Arie has mentioned, that they set a bar that it's now very hard to meet in certain cases. I've been working on cancer vaccines almost my entire professional life. I started with Steve Rosenberg back in the late 90s. And we published, Nick Restifo in the audience, an mRNA vaccine for cancer back in 1999, Nature Medicine. I was a postdoc in the group. And it took us almost 30 years for the science to actually catch up to understand you need neoepitopes, to understand you need a checkpoint inhibitor, et cetera. When we finally at Moderna got to the point that we could test it, we actually did the less obvious thing from a patient perspective, which is we went to the patients with seemingly the less need, which is adjuvant melanoma, patients whose tumor is resected. And some of them it'll come back and some it won't. And we did that because that's the setting where scientifically it's most likely to prove of benefit. And indeed, we published the results a few months ago. It works in adjuvant melanoma, early stage. Now, I don't know yet if it's going to work in late stage. But what's critical here is first and foremost, I think, that we follow the scientific opportunity and we deconvolute the science. I think the next step then is you've got to find a way to make sure that all the stakeholders are around the table. And I think that's where a meeting like this is useful because blueprints or paradigms of development. In this country, we're very fortunate, and although in the current political climate, we often shy away from talking about it, we're the world leaders. I mean, I'm an immigrant to this country for this very reason, because we have the unique opportunity where we have a civil service like no other country. Trust me, I've submitted files in other countries. We have an NIH like no other country, and we have a commercial set of ways of doing things that has enabled progress that no other country has. And I'm very proud of that. I think it's up to all of us, though, to preserve that ecosystem to enable continued progress. And frankly, the thing that scares me the most today sitting up here is not regulatory, it's not finance, it's not the clinical perspective. It's actually the political environment in which we operate.
So Peter, in terms of what was just said around, sort of this remarkable regulatory paradigm that we have in the United States, the biomedical infrastructure, the commercial infrastructure, what do you think is holding us back? Or do you think we're fine? Are we in the best of times, worst of times, everything's great? Where are we on the weather?
I think we're in kind of the middle of times. The issue here is I think we're in a place where science is advancing very rapidly. I think we're on the cusp of addressing certain diseases more efficiently. Some, I think, are probably 5 years away. Some are 10 years away in terms of getting there. Some of them are things that we will see, I think, come into fruition in the next 5 or 10 years in terms of our ability to manufacture more efficiently. I think probably some of the advances that, I don't want to go back and relitigate the CMC panel, but I think, instead of AI making cute cat pictures up for us online, the AI can actually really do us a lot of help in manufacturing, and because the ability for AI to tease out 40 or 50 parameters from a biologic process, which could never be done before, is now something that's possible. So I suspect the next, if we do our jobs right, the next 10 years will bring us great advances in biologics manufacturing. Again, if we apply AI in the right place, rather than making funny cat videos. That's a place where I think, so we'll have advances in manufacturing. I think we at FDA will try to have to keep up with figuring out how do we keep up with the clinical trial designs and understanding how to keep up with things. We'll also have to probably have the rest of the ecosystem keep up with us, because I think we don't live in an isolated world. We have CMS, the payers that are going to have to deal with this. So if we find ways to use some, to update products more frequently based on some level of evidence, we're going to have to make sure that our colleagues are there with us. And obviously, there's always going to have to be safety and effectiveness up front and center. So I think that's a place. And then I think the other piece of this is just making sure that people actually, know what products, that we get people to participate in clinical trials in a big way. That's been an issue. I mean, we were lucky with the vaccines. People were very happy to participate in clinical trials. Cancer patients, on the other hand, adults don't participate in clinical trials as much as they should. Hopefully there will be ways to help them do that. I think we've seen efforts now to try to help people sort through clinical trials. I think Nancy Myers is here trying to work with this system, True Trials, to try to help people figure out what are valid trials. When you look at clinicaltrials.gov, it's become a repository for some trials that are bona fide clinical trials, and others that is essentially advertising for people who are doing stuff that's a little bit on the shady side without either investigation new drug applications or without actually real IRBs. So hopefully that will help sort through some of that. But there's a whole ecosystem, and it goes from manufacturing on through clinical trials out to getting reimbursement. And I think each of those issues are going to have to be addressed if we're going to get there efficiently. It's not just, I think you can't just take one of them in isolation.
So you just brought up AI, and I feel like we could play bingo with how many times AI has probably been raised in the last, month or so. It is the topic of conversation, not just its ability to draw cats for us or write term papers for young people, but what is the art of the possible? And I think about agencies such as the FDA, and I think, well, that's pretty amazing what AI could offer in terms of taking off some of the load that exists. But there is that piece of where the human brain, and our body of knowledge comes in, and the algorithm, so to speak. So maybe if we could talk for a minute about what's the promise of tools like AI to advancing any of the obstacles, that exist currently today.
Peter, you are the expert in AI, I see now. How did we get to AI?
No.
Peter, how are you thinking about it already?
I got there because I think in the world of manufacturing, I think it's certainly something that could be used to help us do process improvement in a way that now that we have the combination of many different process sensors that have been developed, you can actually look in real time, right? AI can look in real time. You can continuously feed it, 20 different parameters, and it can probably identify the critical parameters in a way that we could not do previously. And although I only have a few examples that are proprietary that I can't share, there are ways to go about seeing that. But the other thing is, if you ask on a practical level, what can we do at FDA? Well, we get thousands upon thousands of adverse event reports. With natural language processing, we could probably flag the 1% or 2% that really should be looked at by a medical officer, and the rest that could be categorized and essentially best described through AI as, okay, another adverse event report of a given type, whereas some need to be looked at. So I think identifying what actually requires the human touch is something that AI can help us with. The other piece, ultimately, that I hope we can get to is as we have more and more electronic filings, you can imagine that across genetically modified therapies, that across, we can't share, we won't put out there the different information from different manufacturers for their vectors, but you could imagine if we used AI internally, we might be able, across programs, to find trends in safety or effectiveness across different programs using a given vector, much the same way as you could do the same thing for small molecules, et cetera, find the safest or the best drug. So I think there's a fair amount of room here for it to bring benefit. It's going to take a little while, and it probably will also require some application. And in the one part we don't have the best in the federal government is the ability to get really good. Our information technology is not our strongest suit.
But that's pity, because essentially when you think of that, 10 years ago, there were about less than a handful of companies that interacted in cell therapy with the FDA. Today, there are over 600, and the only one who knows all the questions and the problems is the FDA. So if somebody can take all the information, it's just you. We cannot do that because we don't know the information. And therefore, I think that this is something that very powerful that you can use.
Well, and this is where it comes back to the infrastructure that you raised in terms of the US infrastructure. So on the one hand, you could say we've got this gold standard, right, of the FDA to be ready to think about these issues. On the other hand, we are now in a sort of a political football stage, and you need resources. So any thoughts on that in terms of how to equip the FDA? Because you are at the sort of nexus point, and yet I know, Peter, from being on conference calls during the pandemic with you, you had to work out of your basement with a polar fleece on. We were not treating you like the government representative. I said to you at one point, I said, if you were in Singapore, we would have you in a very beautiful, pristine place with anything you needed to be able to do your job well. I would not say that's how we're treating the FDA employees currently.
Well, they're not treating us that badly. It's true. They're not treating us that badly.
Said it got a little chilly, which is why he put the polar fleece on. So.
Yeah, no. I think that the challenges are that, keeping up with technology in the government is a challenge. Why? Because we don't have, our contracting is a challenge for us. Keeping up with having the best of the best in terms of, and this is just one of the lowest bidder is not necessarily going to be the most advanced in the way of those who might know something about artificial intelligence. So it is a challenge, and we just have to work through that. And I don't have a solution except to say, obviously, Congress can provide solutions, but we don't lobby Congress.
So talk about the perspectives of the rest of the ecosystem in terms of trying to, sort of advance these things as quickly as possible to get them out the door to patients. How do you also see the clinical trial infrastructure, for example, where we need to make sure that we're actually providing access to clinical trials to all people, creating opportunities for equity. Similarly, I think when you start deploying, some of this, this is not kind of easy street for clinicians, right? So the care centers where this would be deployed need to have some, kind of things ready. So what do we need to do to get both the care system, the clinical trial system, even more advanced than it is for this? Anyone who wants to jump in?
Yeah, I mean, I'll say it's pretty impressive when you look at places like Boston Medical Center being able to ratchet up and do CAR T-cell therapies. I think we need more safety net hospitals to have that capability around the country. And then when we think of equity in the melanoma space, we often also think about melanomas that haven't yet benefited from the current immunotherapies. So if we look at acromelanoma or mucosal melanoma, there are populations of patients that, and these subtypes, if you don't know what they are proportionally more common across all races and ethnicities. So we think of those that need to be studied more carefully. So I think of equity both in the delivery side, but also the patients that we're serving.
Other thoughts on that?
I'd put a couple of thoughts on the clinical trial infrastructure. So, you know, blueprints are near and dear to my heart as a drug developer, because I've been a developer like you, Ellen, for most of my life. It drives me absolute bonkers. The clinical trials infrastructure is just gets more and more complicated and more expensive every year, and I'm still a bit hopeful that somehow AI will solve it, and there is a real opportunity. But if you think about it, when a patient comes in to a clinical trial, they get their blood pressure and their weight and their labs, and then they go and they have to do all three again, one for the visit, one for the trial visit, and there's a duplication of effort here that just makes absolutely no sense. People are starting to try and fix it, certainly the big CROs. It hasn't yet, I don't think, gotten to the point that makes sense. What I am optimistic about is, and it's really great to see the FDA move in that direction, is thinking about real-world evidence, synthetic controls, and novel statistics, because I do think in that realm, as we capture more data in EMRs and as the analysis of that data becomes more sophisticated, I think our ability to draw scientifically valid inferences, not just with the usual randomized one-to-one controlled trial, but actually much more sophisticated approach, leveraging that, become relevant. And finally, there are settings and trials in which real-world evidence make a difference. Take a hypothetical. Let's say you have a pandemic, and there's a vaccine, and it works, but it wanes in 6 months. And you want to know whether you need to give a booster shot. And you need to design the trial to prove it. Well, you can't really design the trial to prove it, not in time. By the time you have the result, it's too late. But a country that actually has the health infrastructure did that in real-world evidence, Israel, and showed the rest of the world that indeed it does work. And lo and behold, we all got our booster 6 months later. So there are settings, it taught me there are settings where real-world evidence is not just valid, it's sometimes the only valid thing to do when you take into consideration the time and cost. And it is scientifically valid in the right circumstances.
Arie, do you want to add to this?
In general, when I'm thinking of clinical trials in the pharmaceutical world or biotechnology world, it's all controlled essentially by the FDA. What does the FDA want? And that's why I design the clinical trial. In order to do that, cell therapy is different than any other molecule to be approved. There are so many ancillary medications that we are using that maybe are approved for other indications, but now are not necessary for, it's not the main product, but the ancillary products. You know, even the Cy/Flu, the Cytoxan and fludarabine, and you know, multiple other medications are all important right now as lymphodepletion. So is lymphodepletion part of the main therapy? No, but this is the standard of care. But there are other medications that are coming because the future is combination therapy. You will need more combined therapies, and the question how to bundle all of them together and not to say, oh, this one is approved for X. Now, in order to use it in cell therapy, you have to approve it in that setting. So now, and then you will come back to the cell therapy. So I think that this complexity need to come and be sent a clear message to us of how the FDA looks at it. And that's what we are dealing in several of our companies and need to understand, is it a new drug or not? My personal, I would say frustration, but I think what I would like to see is that CBER and CDER are working together as a group and coming up with these solutions, because you cannot work on one arm or the other. This is a combined effort of both agencies.
So I I gave Ellen the mic for a
I don't know
minute.
if
I
it's
think
an
Ellen
active
wants
one.
to add to this
It better
conversation.
be not active because I'm going to say things that may not be popular. But the truth of the matter is Rob Califf, Rick Pastor, Peter, and others at the FDA have said you do too many stupid trials. And a lot of the trials are not meaningful, and everybody thinks FDA wants certain things that they don't want. Sometimes it's easy. We just did Pragmatica, a simple trial, and getting the cooperative groups to agree on one endpoint was very complicated. So I guess the question is it clear what you want, or are people making the assumption that you want everything and therefore make it more complex than it needs to be, and more expensive and less useful?
Are you asking Peter?
I'm asking Peter.
Yeah,
Because
well,
they're blaming it on
I think-
FDA, and
I
maybe
think
that's
there are two questions here. Does something work foundationally? And that's, I think, one of the issues that's not such a big deal sometimes, because many of these therapies, phase 2 data can show you that they work, because there's such a, I mean, if they're not working gangbusters, you go back to the drawing board to try to make them work gangbusters. The question, though, is that when you have something that's working well, can you make it even better? And I think that's what you're asking for, is how do we go from understanding something that works well, and can it be made better? In other words, something that's got, you know, it's giving complete responses in 55% of people, but you want to get it to 65 or 70% of people, and how do we make that next step without going back to square one of a full development program? That's what I think you're asking here.
Yep.
And I think that really requires us, we may need to have some workshops on how you actually get there with products, in how you evolve in a way without going through a whole new development program. You still need to be able to, because I think we need to have some data to say that, because you're going to want to, you're probably going to want to charge something different for the two products, perhaps. You're probably going to want people to know that there is some difference. And how do you document that?
So, I can't resist, because I think one of the most interesting challenges is going to come up with the personalized cancer vaccine. So we just showed in a randomized phase 2 for adjuvant melanoma that a 34 neoepitope personalized cancer vaccine can actually reduce recurrent-free survival. Phase 2 study, but almost 50% effect. Now the company is going to go ahead and prove it in phase 3. Why 34? Well, that's what I could pack in comfortably. Why not 50? Now they're not going to want to change it, so the phase 3 will look like the phase 2. The choice of neoantigen is based on a computer software, NetMHC, or a version of it, that was NetMHC version 3. There's now version 4, and tomorrow there'll be version 5. Okay, you'll run the phase 3 trial, maybe with version 4. What happens when version 5 comes out, right? And that's going to be software updates, and what kind of evidence will you need once that vaccine is approved to go from 34 neoepitopes to maybe 50 or 60 if I can, right? So these are going to be interesting questions.
Interesting question. I mean, I think the key thing, I don't know if some of our viewers in the audience probably want to say, is they want to make sure that we're just, probably the most important thing is, it's a little bit like if you drive a stick shift. You just don't want to be rolling backwards, right? So that's bad, right? So you want to make sure that at least you're not going backwards with what you've done, and that the safety is not going in the wrong direction. So I think those are the kind of the key parameters that are kind of like the backstops. You can't be somehow losing effectiveness when you think you're going forward, and you can't have new safety concerns that are dramatically different.
So with a vaccine, that's good. The safety is going to be easy. I'm not worried about that. I think the real interesting question is going to be, and I'm no longer part of Moderna, so this is just me as a private citizen curious. Will you be able to show, based on a single-arm study, relative to your other controls, that you're not rolling backwards? Otherwise, you get to the point that was alluded to before, it's going to take you an impossible study to prove you're not rolling backwards.
Well, that's the thing, Ellen, your point was really about the possibility of rolling back. And if we all go back to our memory when we first learned on a stick shift, it was terrifying when you were on a hill, because it was like, oh, no, I can't do that. Like, it's going to go backwards. So are companies doing unnatural moves thinking, that you need something as well?
Yeah, well, I think this is why there's so much talk about real-world evidence, is because I think if you could show that you're safe and you're not rolling backwards, is there some way that we could let things, collect data while rolling forwards in the real world. I don't have the answer to it, but I think it's certainly worth, we're being asked to think about how that could happen, because that may be the only way to get some of these answers without having to go back and do really large trials that people aren't going to enroll to anyway. That's part of the problem.
So Arie, go ahead, and then we'll go to the mic. Start taking
In
some
my head,
questions.
I'm looking at the title here, and next horizon of cancer therapy.
Yeah.
And you're talking about cancer vaccine. So I think as the FDA looks at it, there is the adjuvant therapies, which we are making great progress, and each of us reading the Novartis, the Nature paper for Memorial Sloan Kettering, or Vinod's paper, and Eileen, and what you are describing now, we can see how minimal disease will be treated, and you were talking about it many times, minimal disease in lung cancer and in other diseases. That's okay. But most of the patients today, not in 10 or 15 years from now, are dying for advanced metastatic disease. And I've not heard anything kind of prepared. What's out there for there, or what are we looking at potential breakthroughs in that direction? That's kind of where I'm looking at it. The vaccine, we all sit here and understand. Where are we heading with kind of the majority of solid tumor advanced disease?
So Arie, what do you think the answer to that is from your vantage point? What are we doing?
Me?
No, I said Arie. Poor Mark's like, no.
I'm
not
asking
me.
the question. I'll answer. No, I have my own, but where are you heading with?
It's where you're heading, which is probably in being able to now using, the place where CRISPR-Cas9 can be used most effectively right now without getting us really concerned about what it's going to do when let loose if you gave it directly to a human in their vein is by using it on cells. And so you can make many different edits. You can make cells now that have a whole new receptor complement, so you can potentially, I guess, maybe that's where we're going to see this with people finally going after solid tumors, where you can now reprogram to get multiple antigens and to even put safety checks in to prevent off-target effects on normal tissues. So I think we'll see where that can go. I don't know that we've seen how far that can be played out now. I know where it's being worked on, but we'll see when it's in its application. But I think it's a really exciting time because of what can be done in that setting.
Go ahead.
I would weigh in that there is a lot of work being done in the PD-1 refractory melanoma setting with mRNA vaccines, with cell-based therapies, so more work to do. But even we're funding some early stage work with CAR-Ts and NK cell, advanced approaches in metastatic refractory melanomas. So.
Either of you want to take a swipe at your question, Arie? Where do we think that's going?
I think it's all, at the end of the day, boiled to accessibility. You cannot treat tens of thousands of patients if it's not accessible, if it's not off the shelf, if it's complex, it will not get to the most population. So we have to work on agents that will be kind of off the shelf. Clearly, cell therapy is heading that direction, but it's still not approved. It's, obviously without the allogeneic approach or approaches like that will open the door for multiple more patients to be treated rather than one patient at a time.
I do want to come back to that topic of access, because I think that's an important one as we think forward about health equity and looking at the healthcare system that we have, maybe the healthcare system we dream of, where we all would have access and all physicians you go to would be, at a place to be able to deploy some of these. Let me go to you over here at the mic, and if you want to identify yourself and ask your question.
Sure. Lana Shu, I'm head of regulatory for KITE. I have a question for Dr. Marks. Given the complexity of this science fiction becoming reality, this is innovative therapy, do we see that the agency will be partnering with industry to do more training, workshops, so that we can exchange ideas in real time? Because during my time at the agency as a medical officer, I would write a guidance and maybe 3 years later it gets published. But if we do this in real time, then industry can express what are the forward thinking that they're experiencing right now, and agency can tell us, what are you thinking at the same time, so we can learn from each other, right? So do we see that as a part of the planning, going into the future? Because if we're waiting for guidance documents to come out, that might be too late, right? 3 years, 4 years later. And then we're looking at the agency maybe a little bit more ivory tower in some of their academic thinking, right? Versus industry has moved forward to think of new endpoints, as you're moving into more frontline. Does always that same endpoint that's been used before, is that really, truly fitting for CAR T therapies? So those are the kind of things we want to open dialogue with agency, but yet it is very hard to have that dialogue when everything is written back and forth in the written response, right? And we don't really know when we're reading the response truly what the medical officers is thinking or what the biostatistician is thinking, right? And if we want to propose an innovative way of doing the standard of care arm, right? So if we want to say, can we leverage real world data, right, to be able to not having to recruit patients into the standard of care arm, because patient resources are really precious, right? And to be able to propose some of the innovative ways, we need to understand where the agency is coming from and how accepting they are in those innovative thinking.
Did you say that you also worked at FDA?
I did.
At some point? Okay.
13 years.
Okay, got it.
Right, so there were about 5 questions packed.
I know. So I
there.
think the essence of your question really is, how do we make things happen more swiftly? And I think it goes back to what, Arie, your prescription for the blueprint, which was really about collaboration.
Well, no, I think so. Let's kind of I'm going to unpack some of what you had there. So some of this is a very practical thing which happened during the pandemic. Some of this, let's take care of the 2 pandemic-related things. One bad, one good. One bad was that because everything got so busy and we were so short-staffed, we got to doing a lot of written responses, which industry hates. I get it. I hated it when I was in industry because it's like reading. It's like going to Delphi and like, oh, what does that mean? What does the word "the" mean there instead of and you'd sit there and you'd say, "What? No, seriously." I mean, I remember sitting in that. It was back then. It was Ramada in the Nada Hilton outside of the Park Lawn building, with a team trying to figure out what like 4 words meant from FDA in a written response. And that was back when we didn't get as many written responses. So I get the point about written responses, and we're trying to move by staffing up to reducing the number of written responses and increasing the number of real-time interactions. And I think the communications pilot that we're going to run in rare diseases is probably going to show us that just about every disease would benefit from real-time communication. So that's that piece. The guidance issue is one which goes back to something that Arie mentioned, which is that we are trying to take a page out of Cedar's book, which is trying to do guidance in much more real time. Because you're right, serving up stale guidance isn't really very helpful. And so trying to get them out, shorter guidances that get out of the agency, in reasonable periods of time. So you'll probably see us trying to do that. And then this issue of, can we work together better with industry? You'll probably look for RFIs to come out from the agency, from CBER, because I think we're going to look for how we can work better with industry to address the key issues. Because just like this issue of AI, I think the only way we're going to get to address some of these things is if we work collaboratively around trying to understand what the problems are and then address them. So lots of good questions there. And I think I take the approach that we can work collaboratively with industry to try to identify and work on the problems and still be regulators, right? We still can make sure that we meet our standards and that we hold ourselves accountable. And that's to me what being a good regulator is about.
So Peter, there was a question here from the audience, the hybrid audience, in terms of China is making significant progress in cancer cell and gene therapies, including innovative trial designs. Are there opportunities to learn and inform approaches here? I would add to that, are you at the FDA collaborating with other international regulatory bodies on that particular issue?
We're trying to work with our regulatory colleagues around the globe to see how we can most efficiently review and most efficiently have essentially convergence around what the regulatory requirements are. I think we have to have a little contrarian approach here. I'm not so worried about China. I mean, they have a lot of CAR T cells going on, but I'm not sure whether they have the seriousness in the science in some of they have some very serious companies. I don't mean to offend anyone, but I think we have some very serious companies in the United States that will give them the run for the money, and I'm not sure that's what worries me, as much as our ability to, around the globe, make it easier for companies to be able to have the kind of global equity, even in low and middle income countries, to get those folks access ultimately to cellular therapies.
Go ahead, Arie, and then we'll take this gentleman over here.
Stop telling you one story as we are talking about China. So Zach and the team here with us, second time we came to China, they invited us to Beijing to go in front of the FDA to discuss potential approval of a CAR T in China. Well, we got the data, the information about the meeting. The meeting was started at 8 o'clock in the morning till 3. So it's 8 hours meeting, and we said probably it would be 40 minutes or so with the FDA. Well, there were about 250 people in the room from the FDA, and we, a team of about 4 or 5 of us, working with the FDA for 8 hours. And we came back and said, wow, if we get 45 minutes with the FDA, we are so lucky, and we are working. For 8 hours, we was there with rigorous discussions about which medication, what to do and how. So when I'm looking now, since then, the learning curve of the Chinese regulatory authorities is just absolutely unrecognizable. And you can see the information that we read about the product that's coming out from there, or at least in development.
Right, and I think the issue there that's probably important is that their focus on manufacturing is probably something that may give the United States CAR T cell products a run for their money, because they're trying to bring down the cost dramatically.
Yeah.
All right, we would love to hear your question.
Sure. Dr. Brian Kaufman, I'm the Chief Medical Officer, Executive Vice President of the nonprofit CLL Society. CLL is one area where CAR T hasn't quite yet lived up to its promise, but I think in a lot of ways cellular therapies are a victim of their own success, and as we all know, trials are for patients, not the other way around. How do we make these trials more attractive? We've all talked about that. Can we have control arms? We touched on this, but I'd just like the panel to talk a little bit more. Or can we use historical controls? Can we what kind of control arms can we do so when we have a standardized, like in CLL, the standard of care beyond, if you're comparing to CAR T, if you have double refractory disease, isn't there. So what do we do in these kinds of cases? How do we move the things forward? How do we and in some cases you have a standard of care that's so great, if I've already got something 70, 80, 90% effective, why am I going to try anything different? So these are issues. They're different issues, but how do we make these trials more attractive to patients? How do we look at those comparator arms?
We'd like to take that.
Well, as the one currently not involved in any clinical trials, it's easiest for me probably to take. My sense is, and this is what I was alluding to before, I do think that if you look where the world of data is moving to in terms of integrating electronic medical records much more deeply, we should be able to supplement control arms with a level of synthetic controls and move to a statistical framework that allows us to assess scientific truth in a way that's a bit more modern. I'm hopeful that the field will move into this. I know there are some instances where FDA is already looking at that, and that's what I've been advising my companies when they're looking for ways to do creative proofs of pivotal trials.
So I think I'm going to have to do one quick wrap up and then turn it over to Jeff to close us out. All right, so we'll do just a lightning round. what do you think is the number one obstacle that we need to focus on in whether it's a blueprint or a playbook or something to advancing these therapies to get us to this next horizon? We'll just go down the list.
Started with me. I think continuing the scientific and technical innovation, I think, is the main obstacle. And, you know, just to say one word, if I learned anything at Moderna, it's that there's a world of difference between engineering problems and biological problems. You take a engineer and you ask him, hey, can this rocket get to the moon? They'll tell you quickly yes or no, and here's how much money it's going to take. You ask a physician, is this cancer vaccine going to work? I don't know, but let's try it, right? When we're in the realm of engineering problems, I think there are solutions. I think the combination of engineering and medicine here is where the innovation is for me, still tricky.
Yeah. All right.
You know, by now we have so many patients that we have treated several years ago, and they're doing so well, and some of them don't even want to come to have x-rays for follow-up. Oh, we have been there and they're not coming. What we are doing in our companies is bring this patient back, listen to their stories, listen to the family, and you can learn so much from what goes there. And I think lots of the secrets are in that. How you got to us? What do you do? How is the treatment? What, you know? So I think that listening to patient, and again, what I said, collaboration between the different agencies and the different academic industry, that's the future.
Arie, Steve and Alice are no longer here. I think they left. But you've come so far, Arie, from wanting to be the surgeon, just cutting it out.
I started there.
Mark, go ahead.
Yeah, I'll just say there's a lot of unmet need among melanoma patients, so a lot that aren't responding to treatment. So whether it's a TIL therapy, an mRNA vaccine, to all the sponsors as well as all the academics, keep up the work because there's a lot of patients that need help.
Peter.
And for me, I think it's going to go back to manufacturing paradigm that we really have to figure out. I think the more we understand the critical quality attributes of these products and the ability to understand how they're advancing and to be able to advance them in a logical way, that will, I think, make a big difference.
Excellent. And I think once we get through these different obstacles / challenges, I'll take the last piece, which is access, affordability, appropriate deployment so that physicians are actually steering patients to things that are going to matter. So Jeff, let me turn the baton back to you to close us out. Thank you, panel. Amazing.
Thank you.
Great. Well, thank you all very much for your time and shedding light on a host of various challenges and a complicated field here. But I hope that you found today's meeting helpful, in particular some of the steps that were laid out in the white paper. And thank you very much to all the contributors that helped craft that as we think about steps that we can take to shape policies into the future to actually realize on the promises that have been described here today. Thank you very much for joining us, all of you in the room, for everyone that was online. Please look forward for other events that we have coming throughout the year. We have a number of different meetings and data releases planned throughout 2023. We hope that you'll join us either in person or virtually. But with that, I want to thank everyone for participating today, and hopefully we can continue these important discussions to come. Thank you very much.